So we have 5 minutes, so we will start with a quick video on mango mapping thing and the chatShaala thing, and then I think we will start with the first model system. Okay?
Okay, sir.
Yeah. Yes. So yesterday we have seen. Yeah, the stream has started. So yesterday we have seen at the end of the session a quick intro by Drishant, Jay, and me on mango mapping and form related to it, where we can all together contribute to the research effort. So we will see a quick help video around it. For 2 minutes, yes. Is it visible to everyone? Hello, everyone.
Yes, yes,
Yeah, visible.
Okay, cool. Thanks. So this is a quick introduction to a form using which we can contribute data to map mango tree behavior around us. So there are a lot of mango trees around us, and it's a very interesting project to collect the changes in behavior across geography. And if thousands and lakhs of you together collect this data, we can see the patterns of various aspects like flowering, fruiting, and sprouting across geographical conditions. So to do that, we have created a simple form where you can type in your name, whoever is observing, say X, Y, Z, and whichever observation you're making, say it's mango flowering, and you can type in the space. So if it is other, you can write any other description of that particular observation. Say add the location, say it's Mumbai, and you can provide the latitude and longitude by searching for a place here. So let's say Mumbai. I can quickly tap and the latitude and longitude gets auto-populated. And if you're using it from your mobile, you can tap on this location thing and it quickly adds your location if you enable the location on your access. And you can provide the date and time, and you can, when you're on your mobile phone, you can click here and the camera app opens, as well as your gallery. And because I'm on my laptop here, if I click here, I can quickly access image, and it gets, you can save it if you do not have immediate data log, so you can save it with a number or whatever. Once the internet is back, it gets automatically updated. Right now, because I have internet access, I can directly submit and it gets submitted. So queued records, except those marked as drafts, are uploaded automatically in the background every 5 minutes when the web page is open and internet connection is available. Okay, so that's as simple as that. So hope to see that all of you can easily upload and share more and more observations and be part of this activity. Thank you. Yeah, so that's a quick short demonstration of that, and we will look forward to more and more observations put together, and we'll have a lot of conversations in relation to how nature changes as season changes and how nature is alive around us. And with this quick thing, we are already at 5:30, so we can start the session. Over to Ashutosh.
Okay, thanks, DP.
Welcome all. Welcome to this second day of our annual meet 2020. We yesterday on day one we had a lot many presenters presenting their work. So since March, since the time that we have had the lockdown, all these students, all these STEM players haven't had access to their labs, and so they have come up with alternative ways of doing work, doing their activities, and all of them have set up home labs. And yesterday we saw many presenters presenting all the work that they have been doing in the last 7 months or so. So yesterday we had presenters presenting on the work done on nematodes, snails, fruit flies, and mango mapping. Initially, we also had many of the cubists sharing their experience of cube, of how cube they find it as a different experience than what they find in the classroom. And yes, we also had nice discussions after every presentation. More specifically, the fruit fly discussion, the discussion on evolution went really long. So it has been a good day yesterday, and we had very engaging discussions yesterday, and we are looking forward to more such activities in the days further, including today. So now that we have set the tempo, so let's have our first presenters for the day. So today we begin with the group of butterflies, and we would expect the presenters, those who have worked on butterflies, to present their work. So they would have their video to show. So over to Krishna and Siddhi.
Wild butterflies. Beautiful, diverse, happiness, healthy ecosystem, colorful, and pollinators. Apart from this, they are also important as a component of food chain, as a pollution indicator, as an indicator of climatic changes, and various other roles. So, what are we doing with butterflies? Identifying the butterflies around us, identifying their larval host plants and nectar feeding plants, observing the different life cycle stages of butterflies. Objectives: observe and study plant butterfly interaction, conservation of larval host plants, conservation of butterflies. How we identify a butterfly? We take a clear photo of the butterfly, note the name of the plant on which we spotted the butterfly. Using common search engines like Google Lens, we try to find the name. While using Google Lens, we can see similar butterflies to the one we have. Then we look through websites. We usually follow ifoundbutterflies.org. Next, we look through some reliable articles on that particular butterfly, its banding patterns, its color combinations, spots and lines on its wings. These are some of the butterflies that we identified in our home labs.
Scientific name Medusa procris. The common name the commander. Scientific name Graphium agamemnon. Common name tailed jay. Scientific name Thirumalai limnaceae. Common name blue tiger. Scientific name Cirrhotra thais. Common name Tamil yeoman. Scientific name Mycalessis perseus. Common name dingy bush brown. Scientific name Euthymia hubeneri. Common name the common foray.
Scientific name Euthalia econde. Common name common baron. Scientific name Papiliodemodocus. Common name citrus swallowtail. Scientific name Castalius rosimon. Common name common parrot. Scientific name Urema hecke. Common name common grass yellow. Scientific name Tanasia lepidia, common name grey count. Scientific name Rathindra amur, common name the monkey puzzle. Scientific name Lembric salsala, common name chestnut bob.
Scientific name Hypolimnus bolina. Common name common eggfly or great eggfly. Scientific name Euploia cor. Common name common crow. Scientific name Gemmites lacteata. Common name milky cerulean. Scientific name Michaelensis minius. Common name dark branded bush brown. Scientific name Gemmite selino. Common name common cerulean. Scientific name Parandica aglaea. Common name glassy tiger. Scientific name Neptis hylas. Common name common sailor. Scientific name Dilias eucharis. Common name common Jezebel.
Scientific name Junonia hedonia, common name chocolate pansy. Scientific name Melanix leda, common name common evening. Scientific name Papiliopolites, common name common Mormon. Scientific name Leptosia nina, common name wandering snowflake, also called cyc butterfly. Scientific name Tagiads ghana, common name snow flat. Scientific name Parthenus sylvia, common name Sapiatry clipper.
This is a very interesting interaction we observed between the blue tiger butterflies and Crotalaria plant. In this video, we can clearly see what a blue tiger butterfly is doing sitting on a Crotalaria retusa leaves. This is a photo of the leaves of Crotalaria plant after the interaction of blue tiger butterfly. Why such interaction? From reference, this is due to the presence of pyrrolidine alkaloid in sap. This helps the males for successful countership.
Host plant Albizia chinensis and Cassia fistula. They are found flying close to the ground. Life cycle of common grass yellow. First picture shows the host plant Albizia chinensis, egg on Albizia plant leaf, and the caterpillar stage, pupal stage. 4 images show that different stages of common grass yellow. Egg laying on Albizia plant, white color eggs on Albizia plant leaf, long green cylindrical large black headed caterpillar stage, and pupal stage.
Cyc butterflies. Scientific name Leptosia nina. Common name cyc. White butterflies with prominent black spot on forewing. They are mostly found near grasses. Life cycle of cyc butterfly. They lay egg on cardamine plant. When egg hatches, it gives a caterpillar. From caterpillar, I am going to become a pupa. Pupa attach with silk thread-like structure on cardamine plant. From pupa, I am going to become a butterfly. Finally, I emerge from pupa.
GCMS analysis is an advanced type of phytochemical analysis. Here, GCMS analysis was done to identify the compound that was responsible for the survival of Psyche's caterpillar on cardamine. For preparing sample for GCMS analysis, first I am collect fresh leaves of cardamine and oven dried. After powdering the dried material, it was mixed with methanol. Methanol was used as an extraction solvent for GCMS analysis. After filtering with the Wattsman number 1 filter paper, the sample was injected into the GCMS instrument. A sample volume of 1 ml was injected into the instrument. Within the instrument, the sample starts to separate by heating. Then the separated compound moves through the column towards the mass spectrometer. Helium gas was used as a carrier gas. Then, mass spectrometer identifies the compound. From the GCMS analysis, 23 phytocompounds are obtained. Glucosinolates are defensive secondary metabolites of order brassicales. In plant, glucosinolates are co-occur with myrosinase, which can hydrolyze the glucosinolate. One of the compound called benzyl nitrile was identified through the GCMS analysis, which was the hydrolysis product of benzyl glucosinolate. Benzyl glucosinolate is one of the common specific compound of Pyridae family, that is the Psyche's family. In plants, glucosinolates and myrosinase come together only when the plants start to damage. This result in the hydrolysis of glucosinolate and the subsequent release of isothiocyanate. Isothiocyanates are toxic to most of insects. In the case of Psyche, their larvae redirects the hydrolysis reaction. Redirection results in the formation of nitriles instead of isothiocyanates, and the redirection process is due to the protein larval NSP, that is nitrile specifier protein, which was present in the gut of larvae. When larvae eat cardamine leaves, benzyl glucosinolate in the leaf was hydrolyzed into benzyl nitrile due to the combined action of myrosinase and the larval NSP.
Developmental stages of caterpillar of common Mormon butterfly. These are different stages of caterpillar. We can found on curry leaves plant, lemon plant and sweet lime plant. Here we can see caterpillar eating leaves, and also there are fecal matter of caterpillar. Caterpillar is not moving, not eating and not producing fecal matter now. So from this stage, caterpillar will become a
The red-pierced butterfly is small in size of 3 to 3.5 centimeters. It is always identified by its orange color band on the hindwing. Dorsal and ventral wing patterns are different. Dorsal wings are generally attractive and meant to attract mating partners. Red-pierced has scientific name as Talicada niceus. It has kingdom Animalia, phylum Arthropoda, class Insecta, order Lepidoptera, family Lycaenidae, genus Talicada, species T. niceus. Kalanchoe pinnata is the host plant for the larvae, and Videlia cainensis is the nectar-feeding plant for the adult butterfly. The butterfly goes through 4 stages, starting with the egg, then to larva, pupae, and then an adult butterfly. An unique form of defense mechanism is seen in the larvae stage of butterfly. The larvae does not feed on the entire leaf, but rather it burrows itself inside the leaf and only feeds on the middle layer of leaf. It spends most of its time inside the pocket of the leaf.
Goof-ups. While starting the identification of butterflies, we were not referring any reliable published articles. We just searched with the help of Google Lens to.
After this data collection, it is found that butterflies are specific to some larval host plants. We spotted most of the butterflies on their nectar plants. only a few butterflies were seen on the larval host plants. Even though we identified many butterflies, the number of butterflies in each group is less. Some butterflies which are present in one locality are absent in another locality.
How do butterflies select larval host plants? Is it using any organs or tools for that? What are the conditions of a suitable larval host plant for a specific butterfly? Presence of what in the plant is helping the butterfly to identify the plant? Is the presence of larvae becoming a benefit for the plant in any way?
Okay, thanks for this very nice presentation. I get to see that you all worked on a lot many butterflies over here. So we were just looking at the slides and so many butterflies that you identified. And towards the end of the presentation, we saw that there were so many collaborators. So obviously, lot many collaborators working together. It has been. It seems that it has been possible due to that. So this very elaborate presentation that you had, many over here had lot many things to ask. We are all curious. So one thing that all of us here commonly are asking is that when you were identifying these butterflies, how do you go about identifying here? And I would want the presenters to shed some light on that. So Anusmika over here says that there is some difference between moth and butterfly. Butterflies usually rest with their wings closed, while moths rest with their wings open. Butterflies generally gather food during the daytime, while moths are seen more at night time. Also, butterflies are more colorful than moths. So, what are your views as presenters on what is being said?
And moth, that the main thing is that we mostly see or spot butterflies during day, whereas we spot moths at night. They are more active at night actually. Then the one thing Anusmika termed was that have colors and moths doesn't have, but that is not true. Butterflies are more vibrant, that they come with little more bright colors. They are also butterflies with dim colors, so we can't tell totally that it is color and it is no color.
Even there are butterflies which are like brownish or something, like moth color.
Yes, we have a lot of butterfly with brown color, and when they are sitting in the soil and all, it's hard to tell.
Hard to.
Yes. So this night thing and morning thing is one major thing that we can tell. Then their wings also, when these moths are resting, the wing position is always open, whereas butterfly tend to they close. The position is different. The resting positions.
And you want to say any other phenotypic characters also?
Yes, the major thing that we identified, after all seeing this video, I saw that most of the question was how we identified these butterflies. So we actually started this from this lockdown period. So for starting, we just out of curiosity, we started posting photos in Cube. But when we started doing that, we came to know that it's not an easy task. Finding a name of a butterfly is really difficult. So first and all, like every startup person, what we did was just we depended on Google. We depended on Google links completely. But when we came to do spot more butterflies, take their photos, we came to know that we post the photo of a butterfly with different names, then same butterfly with different names with different photos, like that. So we came to know that the method we are following is completely wrong. So the color doesn't tell in everything about a butterfly actually. So what after that what we do is first like we do it like a stepwise. So first we just use Google Lens. That is a easy search engine so that we can see similar butterflies with what the photo we has. So we use that, then we get many similar results. But we then search for the names and we know the plant on which we saw it, the spot the plant on which we saw that. Then we do is we check websites, but websites is not completely, we can't depend upon them also. So we look at references. If we are using Wikipedia, in the down part we can see reference. So we look out this reference, and then we also look at published papers and all. So these are all available. So this is how we did it, and I can show it with an example. Vaibhavi, can you please? Oh yeah, okay. So this is a butterfly. This is like a blue tiger. So I think many of you have seen this butterfly, but these three photos is not of a single butterfly. These three are of three different butterflies. So when we look at this, we don't get that which name. It is very difficult. So this photograph also, we can only identify their color as black and blue. But when we look, I'll tell that the first photo, it is of glassy tiger. Okay, this glassy tiger is actually dark brown in color. What we observe with our eyes, it's dark brown color. And when we post photos also, many of them in Cube has posted photos of glassy tiger, and it is correctly brown color, dark brown, and not black. But when we look at this picture, we see it as black. The first picture you are seeing is photo of a glassy tiger, and it's dark brown in color. Whereas the second picture is of dark blue tiger, and the third one is of blue tiger. So the second one, if you see, it is completely black color, and the third one is also black. So it's difficult for us to identify which is this. But in the second one, that is the dark blue tiger, the upper side is dark brown in color, whereas in the third picture, the underside is golden brown in color. And also in the second one, it is the streaks and spots are irregular and they are different. So while we are identifying, looking just the color combinations like blue and black doesn't tell the correct species of a butterfly actually. So we just started this work. So we have just started identifying butterflies in our locality. We haven't gone to higher steps like identifying the family, the taxonomy, and other things like that.
So good, Krishna. But then what we want to understand is that how do you get to know? How did you get to know that okay, the underbelly would be of a different color? Was it the Google Lens that was helpful in letting you know about this, or through the references? So how does Google Lens help?
Yes, Google Lens help like Google Lens is a search engine. So it recognizes the image and shows the result, but it doesn't give us a this like it is 100% correct. It is not correct. They also just give a thing that is similar. Yes.
So here we can't completely depend upon a Google Lens. So we want to add some characters that are visible to us like wing color, wing pattern. We can look for that also.
Yes. So once we use like if we are using Google Lens itself, we can just come to an idea like which we can get a result. So what we should do that we should search for reliable articles or any published journals which have the correct details about this particular butterfly. So we actually learned this from our mistakes actually. I posted a photo of blue tiger, then again I spotted, when I spotted I thought it is blue tiger,
but it was not. When Arunima spotted a blue tiger, she posted as glassy. That is when I came to know that it is different. It was brown in color, and so that is how we came to know about our mistakes, and then we started
Butterflies. So, for a beginner, how can we start with identification of butterfly?
So, for a beginner, what you can do that is we learn from our mistakes. So when we are doing it for the first time, you just see we all see butterflies, but we don't even look at them. We just look and see that okay, it is in this color. We have color combination of a black and blue, but all are not same. So we after like identifying some butterflies, only we come to know about the differences. We only observe it clearly then. So what
Yeah.
we so if someone is doing for the first time, what they can, if they really want to know really about this, then they should look at articles, label articles only. That is available in Flipnet like that, and all published articles. So it gives us the details. We use the help
Yeah.
of such articles.
So first we want to observe the butterfly, in which plant these butterflies are coming and how it is flying. Is there any rhythmic movement of their flying and the wing color? We can look at these characters for a beginner.
Yes, we should observe these characters. We
Okay.
shouldn't. Yes.
Krishna, I have a question like why? Okay, so we talked about identification, but my question is why do you want to study the host plant and butterfly interaction? What is the importance of this study?
So, as I told, we just started this of curiosity, but when we begin identifying, we spotted most of the butterflies on their nectar feeding plants. So the details given in that video about the plants along with the name were all the plants that we personally observed and identified the butterfly sitting on. And only like we did our work about like from 8 9 localities. So in all of this, what we found was most of the butterflies we found were on nectar feeding plants, and some of them
So
really wanted to look at larval. They needed to look at the larval cycle, and we were actually looking at we looked at larval host plants, and we were searching for eggs and larvae and caterpillars to find and to study butterfly. And here in this video you can see that we identified a lot of butterfly, but we could observe only 2 or 3 of the life cycles. So in this video, we also told something about this blue tiger and interactions at my home, but I don't see such a big interaction of blue tiger on it. So it is like the plant has been here for 1 year, and it's just 2 of the butterflies come. So we think that it is because of larval host plants. I'm telling this because I did a survey near my house of the of where and all this larval host plant of blue tiger is present, and we couldn't see. So I couldn't find a larval host plant of butterfly. So I thought that these larval host plants are not there, then the butterfly cannot complete its life cycle. So most of the butterflies we found was on nectar feeding plants and not larval host plants. So we wanted to know why that is so. So we think
So
that
to
there is some selection mechanism going on.
Yeah, the butterflies are very selective and they are choosing their host plant to complete their life cycle also.
Yes, the plants on which they feed for nectar, they are not choosing that for their eggs or caterpillars. So only in some of the butterflies that come same, that the larval host and nectar feeding come same, but the other cases it's all different. So we here did an of common grass yellow, then common mormon. In common mormon, they nectar feed on most of the flowering plants in our home, like ixora and some like that. But they complete their life cycle on curry leaves plant and citrus species.
So can we say the if the butterfly is completing its larval host, its larval stage in one plant, and the nectar for nectar it goes to another plant. So can we say that if the butterfly on which plant it is completing its larval stage, it is acting as a parasite?
Yeah, we actually we also have a doubt about that because
Yes.
we want. Hello.
Yes, Krishna.
Yeah, we want to know if the presence of this larvae or egg is good for the plant. That is one of our research questions also. They are selective. The butterflies seem to be selective because they don't have both plants the same. Only some of the species have.
But
So
my question is that only, like, if if a butterfly completes its larval stage in one plant and then going for the nectar for the other plant, then how it is benefiting the plant on which it is completing the larval stage?
about that,
Actually, okay. Okay, Siddhi.
we think that there may be some benefits for the plant too because of this larval stage of the butterfly life cycle on that particular plant.
Okay,
What are the
so
type of benefits like you're talking about, Siddhi?
That is one of our research question too.
so I think because that for searching for that we want to more observation. So then we can find through observation how it is affecting the plant and whether the plant getting any benefits.
Yes, that we actually need to really observe because what we see is just we see that the leaves of that particular plant, then it comes to an inactive stage, then it flews away. So this is what we watch. So we need furthermore something to find out that. So that is our further plan.
Yes. So why we are using this GCM analysis here? What is the principle behind it?
Hello.
Yes,
Can you?
Yes,
Trisha.
yes, yes.
Hello.
Yes, Trisha, you're
Can you hear
audible.
me?
Yes. Yes, Trisha.
Here, GCMS analysis is used to identify which compound that attracts the psyche on. Then I conclude in that. Yes.
Drisha, your
So
voice is not clear.
Hello.
so I think this, yes, Trisha.
So could we have the GCMS slide in
Hello.
front
Can
of
you hear
us
me?
while you say?
Yes.
Hello. Hello.
Yes, Trisha.
Yeah, your voice is.
Hello. Hello.
Yes, we are able to hear you. We are able to hear you. Go ahead.
Hello.
Yes, Trisha, we are able to hear you.
Sir, please ask the question again. I can't hear your question.
So Trisha, we are talking about why we are doing this GCMS analysis here. So this GCMS analysis is a
It seems that Drisha has some network issue. Drisha, try to rejoin.
yeah. I think
Yes,
Meanwhile,
you are
someone
audible
else
now.
from the group could tell us about it.
Hello.
Yes, Trisha.
Can we have someone else from the group answering the same question? Because I think Drisha is facing
Yeah, I will try to answer, and Risha can add to that. GCMS means it is a gas chromatography and mass spectrometry. It is a chemical analysis of the plant, because here she studied about cardamine hirsuta plant and the psyche butterfly. Why the psyche butterflies is attracted to this cardamine plant. So started to analyze the chemical compounds present in the plant. What are the chemicals that are attracting the butterflies?
So why did we not use paper chromatography, and why is it? Do we specifically have to use gas chromatography over here?
Because through this,
Okay, we are again facing issues hearing Drisha. So, Aruna, could you comment? Oh, Drisha.
so I think by using chromatography, it is very difficult to separate all the chemical substance and its measurement. We didn't get a clear idea about that. So I think GCMS is more advanced technique to identify the chemical molecules present in the leaves. So I think she have taken the fresh leaves of this cardamine and tried it and applied for GCM analysis and found something which are the chemicals present in the cardamine leaves.
Okay.
My one question is, why are we using GC only? GC is gas chromatography, right? So is it that there are some volatile components present in cardamine which the butterfly is getting attracted to, or can we use even HPLC, as there could be some liquid molecules which we can analyze using HPLC?
Yes, I think we can. I think GCMS is more advanced techniques. Drisha, if possible, could you explain more about GCMS?
See, GCMS would be used mostly for volatile compounds, and HPLC, that is liquid chromatography, that would be mainly used for liquid molecules. So I'm asking whether cardamine has some volatile components which you are not able to measure via HPLC. So HPLC can also be connected to MS. So you are not able to measure with HPLC MS. That's the reason we are using GCMS, or is there any specific reason for the same? So one aspect
Drisha, your voice
could
is not
be
clear.
simple question we can ask here is whether the plant, the butterfly is going to taste the molecule in the leaf, or it is going to smell it, and why it is getting attracted to the leaf. So that molecule, if it's a gaseous component or a volatile component, then it is appropriate to use GCMS. But if we have a liquid component, then it would be appropriate to use HPLC. So I'm asking, what kind of component could be present to which the butterfly is getting attracted, whether it is using smell or taste.
Yes, this I would like to add that this also is in context with what many are really curious to ask over here. So many are really curious to know, how does the butterfly recognize its larval host, and does it have some form of memory? And also in terms of what Preeti was saying, the mechanism, is there any gaseous component or what? So many of us are really interested in knowing about this. So does the butterfly memorize which is the larval host plant, or something like that? We'll take this up as the last question before we move further to the next presentation.
So I think Drisha can rejoin. We can go to the next question. So how many days this caterpillar takes to become a butterfly?
So could any of the presenters answer this question? How does the fly recognize the larval host? does it memorize this memorize this larval host, which is the larval host plant? Uh, do you know anything regarding this?
Hello.
Yes,
Hello.
Saloni.
Can I add something which I have experienced, like
Yes, yes,
when doing research in red-throated butterfly? At that time, I
yes.
think I've mentioned this a lot of times. Came when the butterfly was laying eggs, like we had a tank system. So in the tank, the butterfly used to be there, and it laid eggs on the host plant. It laid eggs on the nectar feeding plant and the glass. So it is not identifying the host plant and laying eggs over there. Only the eggs which are laid on the host plant, they hatch. Other eggs just don't hatch. That is the thing I've observed.
Okay. So is there no such pattern where the butterfly has a specific plant on which it will be laying its eggs? Uh,
Like you will see them around the host plant much, but then it's not like they will lay only eggs on that
so
particular.
even the larvae that hatch from the eggs would be needing certain leaves to feed on. So is there any specific plant leaves that the larvae can feed on, or is it that they can feed on any plant
Yeah,
leaves?
when you say host plant, it means that the larvae only feeds on that particular plant and that particular part of the plant. Like in case of red curate, it's the leaf. So it feeds only on the leaf.
And, is
Saloni,
there?
you had
Go ahead.
I
Ina.
had a question. Thank you.
Yeah.
You said that the larvae changes into pupae. So, what is the minimum period which a larvae takes to change into a pupae? And what is the behavior which you have observed?
Okay. The behavior wise, they'll stop
Behavior
moving.
of the
They'll
larvae,
stop
of the
eating.
caterpillar. Yeah.
Yeah, so after fully growing, like after a particular size of the larvae, it will start just uh, it will just stop moving and stop eating and stop uh, every little movement of it will stop, and it will just and in 1-2 days it will convert into a pupa after growing like a fully grown larva will convert into a pupa in 2-3 days.
So, which is the inactive and which is the active phase in the life cycle?
Hello. In inactive phase, okay, yeah.
Yeah, which is the inactive phase and which is the most active phase in
Most
the
active
life
phase
cycle?
would be larvae, obviously, because they feed the most in the larval stage.
Hello.
Yes, Shraddha?
Uh, just like Saloni told, the most active stage is larva itself, because when I was observing that of tail jay, I could hear even sounds, like I could see them every day, like they were eating the leaves. That is the main active stage. Then they comes to pupa, it is inactive, we can tell, because then the eating stops and excrete, everything stops.
Okay.
Even Shraddha observed the same, I think, with Sykes. She did with Syke butterfly.
Okay. Okay.
Yes, Krishna.
So, what is the result of your research? Like you have observed the life cycle of, you have mentioned two types of butterflies. What was the result of the test which you were mentioning just now?
Okay, we'll take this up as the last question because we have to move ahead. So, anyone wants to answer what Hina ma'am has asked? Hello, Krishna. Could you hear what Hina ma'am asked?
No, sir. Hina ma'am, can you please repeat?
Yeah, what is the result of your test which you mentioned? That chemical something test which you were talking about. What was the result? What was the conclusion?
That was done by Drishya. So she did that for finding out why does the why does cyc butterfly uh, hello.
Yes, we are
Yes,
able to
yes,
hear
we can
you.
hear you.
Okay, because no, Shraddha and Arunima, everyone who's studying with cardamine observed cyc butterfly egg and larvae on that plant. So when they searched, they didn't see anywhere that the larval host plant mentioned as cardamine. It was like it was written as Cleome. So even we had a conversation about this in the chat shala. So I think Drishya did this study to know that why this particular butterfly is what component on this cardamine plant help this butterfly no help this caterpillar.
Hello.
Yes, Drishya.
Hello. Ma'am, I'm Rishya. I can tell the answer.
Yes.
Hello.
Yes, yeah.
Because I'm done the GCMS analysis to identify the which compound. Actually, through article, I found that the cardamine is for the first time for the first time reported a host plant of psyche. That's why I'm curious about to know which of the compound attracted by psyche on cardamine. For that, I do the GCMS analysis. From the analysis, I found that the benzyl glucosinolate in the cardamine was attracted by the psyche. Psyche belongs to the family Pyridae. There are some common specific compound for Pyridae family for their on their host plants. One of the common specific compound is benzyl glucosinolate.
Okay, okay. So, it's good. We have a lot many questions over here in this very short time. We got so many questions being asked by the participants, and we have very
Ashutosh,
yes.
one more point I want to add.
Yes, ma'am.
Yeah, for a conclusion, because this psych butterfly, particularly ever reported in India, so that's a very novel thing that we did. Which compound is attracted and compound extracted from plant all, and the nature the new thing that cycle and host plant and the compound identification, that is a new thing.
Okay, okay. So we are happy that the students are actually involved in such work. So looking forward to them continuing on this work, and we are really happy that we got so many questions being asked over here by the participants, and a very nice discussion. We would really want to continue on these questions. There are a lot many questions. We can't really take all these questions over here, but all these questions we would certainly want them to be taken up, and looking forward to discussions on our chats, yeah, on our groups. Okay, so we are really happy that we have so many questions to discuss on. So going further, thanks a lot, the butterfly group. We really admire that you have done so much work, and a very nice presentation, and thanks for this very nice discussion. So moving further, what we have next is we would be having the cardamine group. So the cardamine group has something to talk about the butterfly's interaction with the plant. And in addition to that, they also would be describing about the genetic engineering, certain work in genetic engineering that they have done. So we are really looking forward to their work. So could we have Arunima and Manjul going ahead with their presentation?
Comparison of cardamine hirsuta with Arabidopsis thaliana from research point of view. Cardamine hirsuta is a close relative of Arabidopsis thaliana, but it exhibits a morphologically divergent traits from its famous relative, such as in compound leaves, pod shattering, and altered root anatomy and trichome morphology. Become a major tool to uncover molecular mechanisms underlying the morphological variability. In Arabidopsis, the study of developmental mechanisms in compound leaf development, formation of multiple cortical cell layers, or explosive pod shattering cannot be performed in this species. Since then, the evolution of complex leaves from simple leaves of cardamine hirsuta has proven to be an interesting model for several developmental processes, thereby making it a complementary developmental model next to Arabidopsis thaliana.
Where we will find these cardamine plants?
This cardamine plant can be found everywhere.
Yes, I seen this plant in Monar Hills.
And also I went to buy some ornamental plants. Also found some cardamom plants are growing in that pot.
How do you identify this as cardamine plant?
And itself giving a hint. It is a cousin of mustard plant and it is a small plant. I attracted by seeing the seed pods and it having beautiful white flowers.
Such a heavy terms.
A simple way. This plant is having flowers, so this is flowering plant. And when this is at juvenile stage, it has two leaves named dicotyledons. And this belongs to Brassicaceae family.
Wow, it's a kitchen family.
Flower is similar to all other flower as it contains sepals and petals which are 4 in number or tetramerous. Male and female reproductive part is found in a single flower called bisexual.
The arrangement of stamens is tetradynamous. That means 4 plus 2 arrangement. 4 inner long stamens and 2 short outer stamen. Cruciform corolla is the important character of family Brassicaceae, in which the free petals are arranged in the form of a cross.
It is not a single flower. Flowers are seen in cluster form. Actually, it is a raceme, but it looks like corimbos. In the initial stage, all the flowers reaches at the same level. Flower mature and pod will form. Due to elongation of pedicel, the oldest flower is at the base and young root is dry and seed pod will burst out the seed when it reaches to the maturity.
We can see different types of species around us, but the commonly available cardamine species are cardamine hirsuta and flexosa. So, we are in the process of identification.
Rosette leaf of arrangement is seen at the base of cardamine hirsuta. Basal leaves are hairy. Leaves which are present on the erect stem are colline, which are minimum in number. Stamen 4. And seed pod are present parallel to the axis of stem. In cardamine, two types of leaves are present. Basal leaves with entire margin, compound leaves with distinct leaflets.
The life cycle of cardamine is different in different places. Sometimes it may because of species difference. In Ranji, from seed germination to next seed formation, it will take 60 to 65 days. In Kerala, it is observed that 40 to 45 days.
cardamine plants can also be used to study genetic engineering by floral dip method. Why floral dip? Why not any other conventional method? Well, the floral dip method requires considerably less time, effort, and results in greater seed yields. It is simple procedure for transformation from the other conventional method such as tissue culture, which requires time, skilled labor, and relatively expensive laboratory facilities.
The entire process starts from growing healthy plants until it starts producing the floral bud or inflorescence. Next, when it comes to that stage, start culturing the Agrobacterium strain that will harbor the gene of interest. Then, resuspend the cell in a freshly made 5% sucrose and silvet solution. Then, invert the plant and dip only the aerial parts of the plant in the Agrobacterium cell suspension for 10 seconds with a gentle agitation. Then, cover the dipped plant with a plastic cover or wrap them with a plastic film and lay down the treated plant on their sides for 16 to 24 hours to maintain the high humidity. Allow them to grow in isolation and also collect the dry seeds. Check for the transformation. In this case, we used the kanamycin resistant to check for the resistance.
How will you determine germination rate of transformed seeds? For that, I need to determine germination rate of non-transformed seeds. But due to lockdown, I don't have non-transformed seeds at my place. Oh, I found some cardamine plants at my place. Let me help you out there for determining germination rate of non-transformed seeds. But how could you do this experiment at your home during this lockdown? Why not at home? What exactly things are needed for germination? A container, a water, and of course cardamine seeds. Let's try this out. For this, I will need kanamycin antibiotic at different concentration, tap water, and non-transformed seeds. But how are you preparing different concentration of kanamycin at home? For that, first I will be preparing stock solution. Then I will be serially diluting it at different concentrations such as 10 mg per liter, 30 mg per liter, 50 mg per liter, 70 mg per liter, and 90 mg per liter. 20 seeds in control and 20 seeds for each concentration followed by 2 replicates will be used. I am very excited for the results. Then look what I found is kanamycin is not affecting germination rate as 80 to 85% of seedlings have been germinated in control as well as in test medium. Oh, but it's affecting the plant growth. How but? Two factors has been observed. Etiolation, which means bleaching of seedlings, and hypocotyl reduction as concentration of kanamycin solution increases.
That's great result, but at what concentration are we observing the visible effects of kanamycin in non-transformed seeds? Well, in 50 mg per liter, we can see the visible effect of kanamycin in the non-transformed. But I also need to find the rate of germination and growth of cardamine seedling in the presence of kanamycin solution. Well, don't worry, let me help you out in this part too. But why are you only choosing kanamycin antibiotic? Why not others such as penicillin?
Antibiotic resistance in plant. Why not use penicillin? Penicillin kills bacteria by affecting its cell wall. But as we have studied, cell wall of bacteria is different from plant cell wall. Kanamycin on the other hand affects the ribosome of bacteria, and cell organelles of plant cell like chloroplast and mitochondria have bacterial origin. That is, it contains bacterial ribosome. So theoretically, kanamycin should affect plant cell. Let's find out.
GCMS analysis is an advanced type of phytochemical analysis. It combines the feature of gas chromatography and mass spectrometer. For preparing sample for GCMS analysis, first I am collect fresh leaves of cardamom and oven dried. After powdering the dried material, it was mixed with methanol. After filtering, the sample was injected into GCMS instrument. A sample volume of 1 ml was injected into the instrument. Within the instrument, the sample start to separate by heating. Then the separated compound moves through the column towards mass spectrometer. Helium gas was used as a carrier gas in the column. Then the mass spectrometer identifies the compound. From the GCMS analysis, 23 phytocompounds are identified. And the graph shown the GCMS chromatogram of cardamom. Some of the phytocompounds include benzyl nitrile, beta-udesmol, viridiflorol, linoleic acid, linolenic acid, squalane, vitamin E, camphorol, phytol, etc.
Okay, good. Thanks for this presentation. We really like the way you creatively you know showed how cardamom cardamom is an exciting model to work on. So
Yeah, this presentation is really nice.
yes, the presentation was very creative in terms of letting us know why cardamom. So and a lot many participants are also they have questions regarding this. So like you creatively explained how cardamom is a nice model to work on. Many participants are they want to know. You talked about Arabidopsis, and if Arabidopsis is a very good model to work on plant biology, and cardamom is a cousin, and you can work on that. But if Arabidopsis is so well established, then why not simply stick to using that? And we got many answers in the chat as well, but we would want to hear from you.
Considering, okay, Ashutosh sir, answering to that question, I will say that considering Arabidopsis is cultured in a specific condition, they need a specific condition to get like culture, and Caramine is a weed, and considering it is a weed, it is ubiquitous. It will be like found everywhere, and it hardly need any restricted conditions to actually work on. So I think for that reason, we need Caramine because it is more accessible to us rather than Arabidopsis, although Arabidopsis is already set model organism.
Okay, so what is it about weeds? Why when we say it's a weed, we immediately say that okay, this is something that would be very easily available, widely available. What is it so special about weeds?
Wheat actually grow doesn't need any particular requirement like any specific amount of care or something. It actually grow naturally. So and everywhere in a like with the requirement of few water like available of the water source. So considering that Carmine is one of the weed that also belongs to the Brassicaceae family, similar to that of the Arabidopsis. So we consider Carmine as a model organism.
And one more point I would like to address that cardamom plant gives us better germination rate within less time, and leaf development studies can also be understood as cardamom has compound leaves while Arabidopsis has only simple leaves. So the leaf development studies can also be very well understood by cardamom.
Okay, okay. And you also talked about this floral dip method. So can this floral dip method be also done at home?
Floral dip method, the basic requirements, the process is very simple. As we need, as cardamom flower buds are dipped in the suspension culture. So it requires lab, but it can't be done because the culture, agrobacterial culture, would be like culture in lab. So not it can be done at home, but as we have done experiment of on the selective marker that we can we chose a Sanomycin as selective marker. So, like the rest of the experiments, we can do that at home, apart from floral uh, dipping method.
Okay, okay. But then if you have that particular requirement, then it's easy to do everything at home, right?
Yes, exactly, sir. And also, like normal process of transforming plants require the process of tissue culture. So the entire process of tissue culture is being eliminated if we do the floral dip technique. So tissue culture needs to be maintained. Like we need to do the tissue culture, and we need to have a like spend a lot of time to see whether the thing has actually going to a seedling or not, and then we are planting it. So and in respect to that, if we are doing with the Karamine, the floral dip technique, what we need to do is like just dip the flower, wait for the flower to produce the seeds. Once the seeds are produced, you can directly screen it and sow it to see the transformation.
Okay, okay, great, great. So, Bivas, one question to you. You were talking about this Arabidopsis, and you were say that cardamom is a weed. So, is Arabidopsis not a weed?
Sir, to be very honest, I'm not very sure about it, but if like what I have heard, what I've studied about Arabidopsis is like it requires specific condition to grow in the lab. So if any of my team members wants to add something.
Yeah, I also think like Arabidopsis is not we can't consider completely that's a weed because it is not available to us and it is native to Europe. It requires special condition to grow and it requires a nutrient medium. So we want to focus on that to grow very well. But in the case of cardamom, it is widely available and very easy to grow. So that we are considering the cardamom.
Uh, Adding to Arunima and Vivas, I would like to say that yes, Arabidopsis is also a weed, but it is native to European countries. So, due to the temperature differences, we cannot find Arabidopsis here easily. And if we try to grow Arabidopsis in a country like India, which we considered it as like which has in which the temperature is higher than that of the European countries. So we can say that we cannot grow it easily here in the soil directly. So we require some lab conditions, and since cardamine, which is so closely related to Arabidopsis, we can consider in a country like India where Arabidopsis is not very easily accessible, we can go ahead with cardamine to study, or we can choose it as a model organism here, which is so easily
Yeah.
accessible. So that is why Arabidopsis is also weed and cardamine is also weed, but here in the soil of India, the temperature is really suitable for growing of cardamine. So
So in Europe, it is a well-developed weed.
Okay, okay. So now you have this floral dip method slide in front of us. So could you elaborate on it as to how by dipping the flower, how can the genes get transferred? Could you just elaborate on that?
uh, I shall start like how I initiated the process, or to initiate this method, first was we prepared an agrobacterium culture. And after preparing an agrobacterium culture, we centrifuged the culture for 10 minutes between 6,000 to 7,000 RPM, and then we resuspended that agrobacterium culture in a surfactant and sucrose solution. So after that, we brought the plants, then we dipped the plant for 1 minute, and we covered the plant for 24 hours in a poly bag, and then after 24 hours the poly bags were removed and the plants were left to grow in a normal condition. And as it started producing siliquas, so the seeds were harvested on maturity.
Okay, DP, you want to ask something regarding this North India, South India? I did not really get you with that question. What is it that you want to ask?
So people are wondering on YouTube, this question was that comparing two different species, one is European, the South, and also someone added that the plants bought from.
Yes, Sarah? Yes.
Sorry, like I was hearing with a delay. So I think one of the participants was asking how can we compare plants in North India and South India. And I think it's got sort of covered when there was a discussion around temperature and its relation to where these plants are available. And I think some reference to Arabidopsis available in Europe and others has covered it. So if the group wants to add anything further to it, like you can please.
Can you repeat the question? I just missed the question.
Yeah, so the participant was just wondering about the comparison that is there in this model system between plants in North India and South India. So I think the participant was wondering about the geographical conditions and how it affects the availability of plants. So that, I mean, I just pasted the comment here. We don't know anything more than that.
When we studied life cycles, so cardamine was also studied in different parts of India. So as Arunima reported that in her in her place, the cardamine completes its life cycle in 40 days. But in Ranchi, like where I studied the life cycle, it showed like 60 to 65 days. So so we are also not very sure on what, so it could be because of the temperature or maybe geographical condition, or it could be different species, because we are also not very sure about the species right now. We are on the, we are like on the process of identification.
Okay, okay, okay.
Hmm.
Thank you.
So Drishtant has a question, but then Drishtant, we would take up this question during the next presentation when we actually. So he has a question related to horizontal gene transfer, and we would Drishtant, we would take up that question once the next presentation is done. Ritik wants to ask, what is agrobacterium exactly? So how is it helping in this gene transfer?
Well, agrobacteria, if you agrobacteria, we can say that it is a naturally occurring vector.
Can you come closer to your mic? Can't hear you.
Okay, agrobacterium is a naturally occurring soil bacteria. So, which we can say that it is a naturally occurring genetic tool. So, we can, if you have ever noticed that there are some plants which produces a tumor, like a mass of cells. So if the plants get wounded, a certain phenolic compounds are released, and agrobacteria are attracted towards that compounds. And what happens is that they have, since we call that a naturally genetic tool that is present in the soil, it has a virulence gene which has a capacity of producing, we can say, like tumors to the plant. So, which is naturally, so agrobacterium is naturally occurring, so why can't we use it even in our labs to study transformation, like floral dip?
Okay, okay. Okay, so we you in your presentation you also showed us this concept map of how your work gets connected with all the different concepts in your curriculum. So could you comment on that? Could you show that particular concept map and comment a bit on that?
Yeah, so talking about the concept map, we have first related it to the taxonomy, because we are talking about how we are identifying and classifying the carmine plant and relating it to the Brassicaceae family. So that as of like the Arabidopsis. Then we talked about the developmental biology thing, because many of us were trying to study the life cycle of the carmine and also the leaf development part. Then we talked about embryology, because we are talking about seed dormancy, and we have tried to put cold treatment to break the seed dormancy and also the seed germination. Then we are talking about morphology, where we're talking in respect to how we are comparing the leaf forming patterns in Arabidopsis and Carmine. We are talking about molecular biology and genetic engineering, where we're talking about transformation method and pluralistic technique. We're talking about ecological interaction. That's in the last part of the presentation, you have seen the psychic butterfly and the carmine interaction, and also in the first part of the slides, where part of Arunima's work, where she tried to pick carmine from different altitudes and tried to pick up the seeds from altitudes and studied that. We talked about microbiology, because we are also interested about using kanamycin as one of the screening method for the transformation, and what is the working of the kanamycin, how it affects bacteria and how it's going to affect the eukaryote system. Then also research methodology, because we are citing for the references and all. We are talking about biostatistic, because we also talk about like what is the percentage of the seeds that is being growing and what is not growing. We talk about evolutionary biology, because when we talk about the leaf pattern in Arabidopsis and carmine, where we like place them. So there's a so I think carmine, Arabidopsis has a simple leaf and carmine has a compound leaf. So why there's a compound leaf? So what is the so there might be some, because they belong to the same family of Brassicaceae. There might be some changes in the genes that has led to the formation of carmine and the formation of the Arabidopsis. So what is the evolutionary lineage? We also tried to find out that. We also tried to find out that what is the seed dispersal mechanism out there, and also the vegetative to reproductive transition, how it is in carmine. So this entire concept map that we are relating to the
So there is one more question. What's our plan with these transformed seeds? So now we have the genetically transformed seeds and we are using the kanamycin as a selection marker. If the genetic engineering is successful through floral dip, the transformed seeds can grow in kanamycin solution. So our next plan is to grow these seeds in the kanamycin solution.
Okay, okay. So could we also say that the seed dispersal mechanism in that earlier slide, that also
Yes.
comes under reproductive biology?
Yes, in the case of cardamom, it has a wonderful seed mechanism because when the seed pod reaches maturity, it will burst out. Or if we just touch the seed pod, it will burst out. It's a very nice to see.
Yeah,
Okay,
I
okay.
would like to add
So
something. Yeah,
so
am I
we,
audible?
yeah.
So like a golf ball, in golf ball there are some indented. So in the cardamom seeds there are some dimples kind of so that will have seed to fly off from the parent mother plant.
Okay. Okay, good, good. So there are a lot many other questions. Many of the participants are interested to know further about the floral dip method and the exact mechanisms of genetic transfer and how does the transformation take place. So certainly we can take up all these questions. We can continue the discussion in our groups. Uh, so because there are a lot many questions where we can have this discussion continued. So really happy to see those questions coming up. And what we would do now, so thanks a lot for this particular presentation, the cardamom group. And we would take a very short break and have a group photo now before we move on to our next presentation over here. So thanks a lot, the cardamom group. I would now request all the participants that are here, if they could just turn on their cameras so that we could have a group photo.
Yeah, sorry to interrupt, but I would like to say there was a slide in the PowerPoint. And Prashant, if you can put that slide again, and I would like to show a picture in which the cubists from Cube Nature Mechanics, they have taken the initiative to study the seed germination. So there's yeah, you can go back. I can't see anything.
Yeah, here we can see the germination of mung seeds. This seed germination is very visible here. I think they started to germinate the seeds in normal water. Kiran ma'am, is it visible to you?
Yeah, is it visible? Am I audible? Yeah. So I would like to encourage their work. Like this work was done in Cube Home Lab, and the main objective is to study how does a seed germinate. So that was a great initiative taken by this cubist. Yeah, we.
Kiran, your voice is low. Could you be closer to the microphone?
Okay, so sorry for that. I was saying that I would like to encourage the work that the initiative taken by this cubist from Cube Nature Mechanics group. So the objective of the study is they were studying how does a seed germinate, and the seed germinate actually helped us a lot in order to understand how the cardamom seed germinate. So that's all I would like to say. Thanks for that. Thank you so much.
Okay, thanks for that. So now we are getting to see the participants turning on their cameras. I would want all the participants to turn on the camera so that we could take a group photo.
Yes, all of you please turn on your cameras, and I think we'll take a screenshot. And all of you also can take screenshots, whoever is available. So yesterday we had 18 people at a time. So let's try and have more today. Yeah, we have 2 people with Anushka. Yes, more people, please turn on your cameras. Yes. Yeah, we have 17 now.
A lot many are yet to turn on their cameras.
Yeah.
All those, even the presenters, Vivas, Kiran, can you?
But it is showing permission, no permission.
So I think the browser would require camera permission. Yeah, for those who are joining from your laptops or machines, I think if you tap on the log right in the beginning of the URL or on the right side of it. Arunan, are you facing any issue? You need to tap on the third icon. The video icon, and it should be visible. Yeah, I think 17 is the maximum that we touched today. We haven't been.
So I think we cannot have access now.
Tell us when to smile. Keep smiling always. Yes, come on, come on. It's 18 now. Just 2 more. Let's make it 20 to 19. Yes. Arun is also connecting. No, I think there's no limit. It automatically arranges itself. Is anyone seeing Arunan and Shraddha's pictures yet, or is it loading for everyone?
Loading for me.
Loading.
What I joined?
Yeah, it's still.
Oh, you can't see.
Yeah, these are still loading.
Voice.
The The biggest goof up of all would be not continuing the work, of course, because we used microscope in labs. Of course, we didn't have access to it in lockdown, but can we do it at home? So now we are learning from our goof ups and we are preparing home labs. The size of rotifer is around half mm, so it can be seen by naked eye. So do we really need microscope? We had made single line cultures for further identification of belloid rotifers through serial dilution, further designing our experiment to prove or disprove our hypothesis. Because of pandemic, we lost all our cultures. Nevertheless, we are starting our work all over again, developing home lab.
Look what I found in Paglopus. Doesn't it look like an organism? Now, what is a Paglopus? You must be wondering. You all must have heard of Galapagos Islands, which is rich in biodiversity. Similarly, microenvironment rich in biodiversity can be found even in an urban jungle like Mumbai. Now, what could this be? These are depressions seen on a manhole lid covering the underground sewage pipes. Well, these are rich in biodiversity, especially in rainy season and at microbial level, and are easily accessible to young researchers. These unique microecosystems are named Paglopus by inverting the word Galapagos. Rotifer were chosen as model organism from this microbial world.
Culturing. Collect soil sample from Pagalapos or a plant nearby. Add it in plastic bottle. Fill the bottle with approximately 250 ml of dechlorinated water. Add 1 drop of milk. Milk will be the nutrient source for bacteria in the culture. so that bacteria divides and rotifer feeds on them. What can we expect in the soil sample? Just rotifers? We can also observe nematodes, parameciums, dirt, algae, depending on the environment of the soil sample.
Sexual reproduction creates every generation this genetic variability that is essential for an evolution. So, does it mean that the animals that lost sexual reproduction or that abandon it or have no sexual reproduction cannot evolve, cannot adapt until we discover what has been called as an evolutionary scandal or an ancient sexual scandal? It's microscopic world of animals, the rotifers. Their body is generally divided into head, neck, trunk and foot, and they are about 200 to 500 micrometers. Rotifer contains three classes in that belloid rotifer are easily recognized by the pair of ciliated reversible discs that is present on their heads. On the dorsal surface of the heads, all species have dorsal antenna that varies in length. The mode of reproduction is generally parthenogenesis. These are the females cloning themselves. Never has any male has been discovered. They exist since millions of years and we found them everywhere. And they are not interesting because they can reproduce without males and evolve without males, but also they can survive in unfavorable conditions. They have more than 400 species being described as being diversified into many morphological forms, all females reproducing without males. So, how do they get evolved? How do they get variability? Because we know it's essential for an evolution. Hey, Rotiver, how are you surviving from last so many years without sexual reproduction?
I can survive in no water as well as by desiccating myself just like dormancy.
So, how you do that?
I have the capability to be in this condition for a long time and then come back to the normal life.
That's cool. Is it your genome still intact when you go under dormancy?
Yes, and Claudia Ricci had done the experiments to study dormancy and variations. Do you wanna know a fun fact about me?
Yes, why not?
Matthew Michelson also found gene of bacteria, fungi and plants in me.
Really? Matthew Michelson of Michelson and Stahl experiment? That's cool. Okay, so if you take bacterial gene from the environment, it is definitely not from the parents to offsprings. So is it HGT, that is horizontal gene transfer?
That's still a confusing mystery for everyone.
Hello all. In order to come back to our original question, does horizontal gene transfer takes place in belloid rotifer? How do we go about proving that? Feeding antibiotic resistant bacteria could be the easiest method. We expect belloid rotifer takes up the genetic material from its surrounding and incorporates in its genome. Antibiotic kills bacteria. Can it also affect rotifer? Questions like these should answer, can antibiotic affect animals? On a larger level, can it affect humans, you and me? Penicillin, the antibiotic to which we all were introduced in school. It affects the cell wall of bacteria. And animals don't have any cell wall. So such antibiotics shouldn't affect rotifer. Kanamycin, yet another antibiotic. But instead of cell wall, it affects the ribosome of bacteria, which is also found in mitochondria, the powerhouse of the cell. 7th standard biology. So if mitochondria gets affected, will the cell get energy? Will the rotifer survive? We can try finding out that what happens to belloid rotifer in different concentration of kanamycin.
other bacteria in the same vicinity. So from that, how are you, like, if you see for
Yes.
normal,
So
for
in
normal,
that,
like normal example, if you
yeah.
see, if we eat something that goes into our gut and it get digested, similarly it will happen with the rotifer. So it will happen with the rotifer. So how can you hypothesize that while they're eating, the bacteria will go or somewhere transform into the, or do the horizontal gene transfer? Then if you are hypothesizing in that way, then every or the other second we will get transformed because a lot of bacteria stays in our gut.
See. Oh, okay, I get the problem. See, HGT doesn't happen in humans. We have vertical gene transfer. We get gene from our, like, there is vertical gene transfer, male and female mate, and so sperm and egg combine together to form a zygote, and that's how new humans can develop, like newborn babies are. So in rotifers, I get your point that why should one hypothesize that while eating the bacteria, the gene is getting transferred. So in that paper that you mentioned, they are actually not banging, they are not concluding that it is horizontal gene transfer. It is their hypothesis actually that it
Now
is horizontal.
they have concluded that it is horizontal gene transfer, and they have backed with cross references. Have you
No,
read that
yeah,
cross
I've read
reference?
that paper. Surely I'll share that paper, Bivas, in the group, and I'll
Yes, I
point
will also
out.
share that. I will
I'll
answer the cross references as well. There
yes,
are cross
I'll
references. Okay, there are cross references in 2020 which describes the entire mechanism of how it does. So just if you can check out, there is a paper from the Matthew Michelson itself, the lab, which says that when the rotifer undergoes the process of desiccation, there's a horizontal gene transfer. So can you tell more about that?
So Saida,
yes,
before you give the explanation,
yes.
because in the last presentation, Drishtant had asked about whether there is a similarity between the floral dip method that we use,
Yes.
where gene transfer takes place from the agrobacterium to the plant, and with horizontal gene transfer. I purposely wanted that to be addressed here because
Yes.
then the participants would have an idea of horizontal gene transfer. So while you are answering, could you answer in a way where even that question gets answered?
Okay, yeah, I'll try. So in cardamine, we are trying to transfer gene of antibiotic resistant gene, same kanamycin resistant gene in the plant itself. And of course, it's not already having it. It is a horizontal gene transfer. So basically, what is horizontal gene transfer, if I may say? It's transfer of gene from one organism to the other, but there is no parent to offspring involved. The transfer of gene is not between parent to offspring. It can be between two different species, same species. It can be between two different organisms. And here we are talking about rotifers and bacteria. So it's quite a different, as in rotifer is an animal, and bacteria, it's like eukaryotes and prokaryotes. So yeah, so that is what it is, horizontal gene transfer. So in that cardamine plant also we are kind of doing horizontal gene transfer, but here the process is happening naturally. That is why it is called horizontal gene transfer. There it is called genetic engineering, because we are doing it. We, like humans, are putting the genes.
So Saida,
Yes.
over there, as was said by one of the presenters, that agrobacterium also naturally transfers the genes. It's that we then use it for genetic engineering. We use that naturally occurring genetic tool, and we use it for gene transfer in the plant. So agrobacterium naturally has an ability to do that, and in rotifer. So as far as what I understand, so vertical transfer is where something goes from the mother to the daughter.
Yes.
And horizontal transfer is something like, I am able to transfer genes from me to my brother or my sister or something like that. So
yeah.
when you say horizontal gene transfer between the rotifer and bacteria, I somewhere felt that it was between from one rotifer to the other rotifer. Is it not like that?
No, it's not like that. So when we were talking about antibiotic resistant bacteria, it is so the paper that we were talking about says that the rotifers are taking up gene from the environment. So so if we put antibiotic resistant bacteria in this rotifer culture, and of course rotifer feeds on bacteria, so they might feed on them as well. So if there is a gene transfer, they can also take kanamycin resistant gene, through which we can prove our hypothesis whether it is horizontal gene transfer or not. Um, so
Saida, question like why there's a need of horizontal gene transfer in rotifer, and another question adding to that is like why we are using kanamycin as such to make to screen, or why not we are using GFP, because I think GFP green fluorescent bacteria will be much more easier to screen.
okay, see, green, we can use green fluorescent bacteria as well. GFP could be um, an option as well. But why we are talking about kanamycin resistant gene or kanamycin resistant bacteria is because we are trying to, as someone I think mentioned in the chat box, that we are kind of trying to relate cardamine and rotifers as well. So they are using kanamycin resistant bacteria, and the question was, can we try the same thing here to prove horizontal gene transfer? Doing the same method to ask two different questions.
Okay.
And
Saida,
why we need horizontal gene transfer?
Bivas, I would want to take a pause over here, because many of our participants who have joined this particular thing through YouTube weren't able to see the initial part of the video. There was some technical glitch. So let me just very quickly explain what we had in the initial part, so that even they would be on the same page. So for those who couldn't really attend the first part of the video, what was being explained is...
Should I play the video again?
Yes, you can try doing that.
Sir, actually there was the audio problem after the video, like some time after the video, so they weren't able to connect to that. So the
Okay, I
YouTube
think
users
so. The best thing to do is
after the video, the starting part.
Yeah, so let me know what should I do. Should I play the video once again?
Better would...
Yes, Sashi.
The issue was after the video was done, right? So Saida or Ashutosh sir can go ahead with that.
So I think there was no problem. Yes, Sashi.
Hello?
Yeah, we can hear you.
Yeah, sorry, I got disconnected for a while. So are you playing the video? Saida, what I was saying is that if you could explain in your own words the initial part of the video rather than replaying it.
Okay. So we started off with goof ups firstly. Yes, Lokesh, please.
So rather than going to that in deep method, so I will like to add that is a rotifer is basically the new organism that is never introduced our curriculum. And one more point I would like to add, that is that why we have started with our goof ups is that owning our mistakes and learning from our goof ups, that is what we have learned from the cube lab. So first of all, we should know what is the Pagalopus and why we are connecting it with Galapagos Islands. So let's continue with Monta.
You must be wondering about Paglopus. So, Paglopus is also known as manhole. Generally, in this area, there are sewage system, which is underground. It covered by lead. There are two depression. So, in the depression, over time, there are collection of dust, soil, water in our doorstep. We have studied in curriculum about Galapagos Island in Darwin theory. So, Galapagos Island are surrounded by water. Similar to that, Paglopus are surrounded by land. So, there are similarity that we can study variation because there are more ecosystem present on the island. So, Paglopus word is coined by cube life by cubist. By inverting the word, Galapagos. Galapagos and Pagolopus both have same ecosystem. Are
So yeah, basically we why Pagalapos and stuff like that. I'm getting questions in the chat box. So it was actually not. If you do a Google search, you will definitely not get anything because this is a term coined by Cube. I would like to say that because why we are kind of coming. Someone muted me, sorry. Why we are coming to Pagalapos is because this is biodiversity at a doorstep. If you would have seen in the manhole, there are two depressions, and over time, like Mamta added, over time there is soil and water collected on it. And it's not about just rotifers. You can see a biodiversity there where you can find nematodes, you can find bacteria, you can find fungus, you can find algae. There is a small ecosystem growing there. So yeah, just pointing out that, and surely there is no restriction of rotifer being found in Pagalapos only. You can find rotifer in ponds, lakes, also in soil, moist soil, dry soil. So basically, this is in last Cube chatShaala when we were discussing about rotifers, I was kind of banging on. Invite all maybe to join us and let's look for rotifer. So what we are doing currently at home lab, because as we added and as Lokesh added, especially in between that, we started the video with goof ups that we didn't continue our work for a long time. So and now we suddenly are starting all over again is because that's what we learn from Cube, owning our mistakes, right? Learning from our goof ups. So and yeah, Ashwati from Fruit Fly Group, they actually started this. They started identifying uh, fruit fly, whether it belongs to Drosophilidae family, by using phone camera. So getting inspired from them, we are trying to search rotifer as well and try to see it with our phone camera. The size of rotifer is around 0.5 mm, that is half an mm, like to 0.1 to 0.5 mm. So it can be seen by the naked eyes, and I'm not denying that it can be seen by the naked eyes. But if you see in the bottle, if you see in the culture, it looks like dust particle moving around. So you cannot really locate whether it is a rotifer or not. So for that we need some magnification or something that could be we can use phone. And surely we have a workshop today on microscopy, so we will get even better ideas regarding how to use our phone in this lockdown as a microscope.
Okay, so Sarah, could you very quickly tell about the similarities and differences between the Galapagos Islands and the manhole lids, the manhole lid wells?
So the similarities and dissimilar. So the first dissimilarity was the term actually Pagalapagos was coined because we kind of inverted the word Galapagos, because islands are what is land covered by water. Here there is in the wells, in the manhole lids, in the depressions there are over time soil and water collecting, and around that there is land. So we kind of inverted the word Galapagos to Pagalapagos, and we didn't it. Actually, it was not as the credit should go to our former cubist. So it's so Galapagos Island is not just one island. There's a group of islands where Darwin actually studied evolution. That there was variation on basis of environment um, in Galapagos Islands, and and that can be replicated in the manhole lid. So if you see the manhole lid, we can see two different environments in both the depressions. Say one is falling under a tree, it is in shade area, and one is in direct contact with sunlight. There are two different environments created. So can there be difference in the rotifers in both the wells? We can try to study that.
Okay, thanks. And so very quickly coming back, so because many of the participants needed an explanation of this initial part of the video, we got back to this. But then very quickly coming back to what was being asked by Bivas. So why is it that when they eat the bacteria, rather than the bacteria getting digested in the gut and all the genetic material of the bacteria getting lost, destroyed, how is it that the genetic material gets transferred, incorporated into the genome of the rotifer? And so like the way agrobacterium transfers the gene and that gene gets incorporated into the genome of the plant, is are there similar mechanisms over here? So could you just very quickly say on that?
Yeah, so I'll try to touch upon that. As in, so in Galapagos, sorry. In this rotifer, when we talk about HGT, that the bacteria, the genes of bacteria is getting incorporated in this. It's not that every time it will eat a food and the genes will get incorporated. No, it's not like that. The paper talks about desiccation, like Devas also added. Now, desiccation, what is desiccation would be a bigger question. Desiccation is kind of a dormancy, we can say. Yes, Lokesh.
So generally, what is the desiccation? As the unfavorable conditions, rotifer is mostly found in moist soils. So as it soil gets dry out, so the rotifers go under the dormancy. That is the desiccation, where the water losses can be seen in the water loss can be seen in the rotifers, which is known as, we say that it is a anhydrobiosis. That is a complete loss of water from the body. From that, the rotifers go under. They were,
Yes, we are able to hear you. Go ahead.
Okay, so they undergo the desiccation, that is similar to the dormancy. So the water loss gets completely there. The metabolic activities that not get completely stopped, but they get slow. So in between that, that drying process, uh, the cell membrane of the rotifer gets break down, or they get shattered, and that their DNA gets broken down. So the as we you must not be you must be similar that the rotifers generally they are good in the repairing their DNA, so that it repairing the DNA. So after they get favorable conditions like moist soil, or we add the water in that, so they start rehydrating. That is the revival of that. So it takes almost 7 days to rehydration, as the experiment done by the Claudia Rishi. In that share, it was published on August 2013. That is a morphological response of Deloitte rotifers. So in that, as they get revival in 7 days, the completely broken DNA starts recovering. So she has hypothesized that in between that, the surrounding DNA, what is as the bacteria gets dies in the surrounding, or some other rotifers also have their DNA in surrounding of that. So they get chunks of that. There is a very they get entry point for the chunks of the foreign DNA in between that. So the
So that is the hypothesis behind horizontal gene transfer. So it is
I have a
so
doubt with
yes,
what
Devas.
Lokesh was saying.
Yes,
That you said that in the case of desiccation, the cell membrane breaks down and the DNA gets fragmented. So if the DNA gets fragmented, how the organism is surviving?
so
but there is
actually by that time, yes, Lokesh.
but the molecules of the DNA are also present, and the rotifers can survive with the broken DNA with the help of the what sorry
Antioxidants.
antioxidants.
And why will it take the DNA only during the desiccation period? What
So
is
it is
the
not
like?
proven yet, Bivas. It is just a hypothesis, and there are many papers, many such articles based on hypothesis where they are just putting forth their hypothesis, and we are kind of in that line, you can say, to prove whether really horizontal gene transfer takes place, because this organism, like, why you asked the question really, that why is
why should horizontal gene transfer takes place, right? So
But I have a one question that okay, I understood that that paper talk about desiccation and the gene or the DNA fragments anyways getting incorporated into the body of the rotifer organism. Then why you're in your experimental setup you're feeding the organism rather than creating a desiccating condition?
feeding the organism, as in when we are, as I mentioned before also, that feeding the organism, as in what the paper suggests by Matthew Michelson, that they are taking genes from their surroundings. So we are kind of putting them in antibiotic resistant culture, so they can also feed on them. Sorry if it got misquoted. I'm really sorry. That's not what I meant. So yes, there could be several cycles of desiccation and revival, and we can try finding out whether it is really without desiccation that this can happen, or desiccation is really important in that process.
That brings an
As per,
important question, like why there's a need of, like you said that whether it will take whether it will happen without desiccation or with desiccation. So that brings an important question that why there's a need of horizontal gene transfer overall. Why the veloid rotifers can't rely on normal variation that it gets from.
so
So,
see, when yeah, okay, say.
so because first of all, we should know why Deloitte rotifers and what are they. Actually, the rotifers are in the rotifer. There are three classes, that is Deloitte, Monogonata, and Cincinnatidia. So in that, the Deloitte rotifers have only females surviving since last 18 million years. There are no males are found yet. So that's why for the generation for the evolution, there is a variation in the we need. So that's what I have
So variation
start.
can be done from the mutation itself
Yes,
as well.
yes. That we do not disagree that mutation can occur, and mutation is random. Um, it can definitely occur and can cause variations. But so as we are saying that we are kind of banging upon the literature as to what work has been done so far, and we are kind of
But
continuing
then though I won't agree that what is the need of the HGT if you're banging upon your hypothesis on based on the, like, there should be a scientific reason that why you are
yeah,
saying that
okay,
HGT is happening, because I will say that the
yeah.
variation that the Bellodina or the Belloida rotifer that is getting entirely from the mutation. Why will I believe
So,
that whether it is getting from
okay,
HGT or
for mutation to happen or to make change in the rotifer, we have, of course, when we were discussing about coronavirus, we had touched upon this, like what is mutation and what are different kinds of mutation. For making a difference in rotifer, for making a variant, the rate of mutation could be very slow, because the mutation is very it is random. It is not like after 20 replication or 30 replication or something like that, then the mutation will occur. There is no time limit as such.
also there is no
Secondly,
time limit, so we can't
yeah,
also deny the fact that it can occur after every 10 replication as well.
okay, yeah,
As you said, the
yeah.
entire process is random.
Let me complete
So
with that.
Vivas,
Yes.
do you have a sibling at home? Do you have a brother or sister?
Yes.
Elder brother, elder sister?
Younger brother.
Younger brother. You look different than your younger brother, right?
Yes.
Yes. So can you totally say that that is due to mutation, or how do you count that difference between the way you look and your brother?
There are many factors that comes in. So there are like the variation that we get from, because half of the sets of the chromosome comes from my father and half of the sets of the chromosome comes from my mother. So there are a recombination
And
happening at the cell division level as well.
Just a
okay,
second,
okay,
Vivas.
Vivas.
Yes, Hina.
Yeah, I just needed to ask a question from Vivas. You mentioned recombination and variation happening. So, don't you think that this kind of variation and recombination would also happen in Deloitte rotifers?
Ma'am, it won't happen because considering Bellaria doesn't have any male, as Lokesh quoted on, Lokesh only quoted. And if there is a variation happening in the Belloid rotifer because of the sexual reproduction, then why would it like take genes from the
There is
other
no sexual
organism?
reproduction, Vivas. There is no
There is
sexual
no sexual
reproduction.
That's
reproduction
what that is what I said. That is what I said. Now my marks whether the similar type of variation will you see in the Beloida rotifers. So I said, as Lokesh mentioned, there is no male in the in the community. Then we can't say that there is similar type of variation we
So,
can get
So
from the
but
rotifers. So
but then we can we bang on mutation to cause variation, because the rate of mutation we don't know. A secondly, there are many types of mutation, and can mute for such a mutation to happen to make a variant is very low. The chances are very low,
how
because
can you say that if there is a variation in the
yeah,
latent gene?
I'll explain. I'll explain. I'm ready to explain because don't worry. Um, so when we talk about mutation, the in the whole DNA is not coding for some protein. There could be a major part could be junk DNA. So if there is a change, there is a mutation in the junk DNA, there would be no variant made, because that's a junk part that doesn't code for any protein. A second would be not one amino acid, not one code is coded for one amino acid. Like there are multiple codes for at least 2 to 3 codes for one amino acid. So if there is a change say if there is a mutation to there is a change in the sequence, but ultimately it's producing the same protein, there would be no difference. There would be no variance occurring.
That is a particular case. So
Yeah,
like if
there
you
is
are
a
saying that
particular case.
there is
So
a particular case, I can.
what I'm trying to say is, for such a mutation to occur to make a variant, the rate is very slow. The chances are very low, and they are surviving for so many years. I don't disagree with your point that there can be mutation. I'm not disagreeing on that, but uh, that for them to have for them to have variants,
So
mutation should not
the
be
same.
only reason.
That
Okay,
means,
I
that means,
understood.
Saida,
Yes.
that means I would like to make a statement to both of you. Like, this is a very strong discussion. One person is saying why variation, the other is saying mutation is ineffective. Saida, that is what you're meaning to say. That the mutation which is
Sorry,
occurring
sorry.
is silent mutation.
Not maybe I'm
It's
And
not.
also
silent.
one more point, and
Mutation is
also
not. This
one more
let
point,
me
side of
let. Let me
Give us
finish.
let us complete one person at a time.
Yeah. I'm not saying that mutation is not at all responsible for variation. No, I'm not saying that. Please don't uh, think that way. Mutation can be responsible, but I'm saying for such a mutation to happen, the rate is very slow. The chances are very less, because there are several types of mutation, and mutation is random. There is no specificity that mutation will occur at a particular time. That it is random. It can happen also. It cannot. It may not happen also. And if it is happening, there are several types of mutation to occur. For a mutation to make a change, it's you cannot really say that variation that is occurring in rotifer is totally on mutation, because there are so many different species. If there is just one rotifer and it is kind of cloning, there should be only one species. There are several species, as we mentioned in the video. There are hundreds and thousands of species, and there are hundreds and thousands of rotifers in the surrounding. So how are we getting that change? Can we really bang on mutation for that? So
Okay. So this is a very strong discussion that we have. So we actually have come to a point where Vivas has a few points to say. Sada is not totally disagreeing on it. So, but she has a few points. So it's a very strong discussion. But we are also strongly falling behind time. So my suggestion is that we would really look forward that if we could continue this particular discussion on the group. So just because I would say here that let's end this particular discussion over here, and let's move on to the next presentation.
Or we can
Please
take
do not
one question, last question.
just a minute, Prashant. So let me just complete with what I was saying. So all I'm trying to say is that even if I say that we are moving to the next presentation, we would really want this particular discussion, very interesting discussion, to continue on our groups.
I will show my experiment for rotifer.
Acha, okay, so you have an experiment to show for rotifer. So just a minute, Aryan. Okay, so this is what you have uploaded.
actually, Aryan as a new collaborator that he's 7th standard student. He's joined a group. He's trying to ask the same question that we are. He started culturing, so I think he's excited about it and he's showing what he's
Yes,
done.
yes, we would want to look what he has to show. So Saida and Biva, so we are really looking forward for you to continue the discussion. Okay, we don't want this discussion to die out. So if you could continue on the group. Over to you, Aryan.
Yeah, sure. We can continue the discussion on group. And yeah, it was very interesting about talking about
certainly.
for evolution and mutation. Yes.
Certainly it was. Certainly it was. Aryan, could you show us what your experiment was, what the work? We are really interested to see that.
Please repeat.
You had some work to show, right, Aryan? We
Yeah.
all are curious to look at it.
Yeah, this is 400 ml बॉटल एंड इसमें मैंने 250 ml डीक्लोरिनेट वाटर डाला है और इसमें मैं 1 ड्रॉप एंड 1 टीस्पून मिट्टी जो मैंने गमला से लिया है और मैंने इसमें डाला है और 1 ड्रॉप मिल्क मैं इसमें रोज डालता हूँ अभी 2 दिन से। So
आर्यन,
this
तुम
is my
कुछ
experiment.
यहाँ स्क्रीन पे दिखाना चाहते हो क्या? हमें कुछ दिख नहीं रहा है। तुम्हारे
Okay,
तुम
yeah,
चाहते
I'll
हो
show you.
कुछ? हाँ, अब दिख रहा है। So अभी you can give your explanation.
Yeah. So मैंने इसमें लिया है डीक्लोरिनेट 250 ml एंड इसमें मैंने डाला है 1 टीस्पून मिट्टी जो हम लोग गमले में रखते हैं और इसमें मैंने 1 ड्रॉप मिल्क रोज डालता हूँ इसमें सुबह। अब ये मैंने 2 दिन से डाला और ये मेरा एक्सपेरिमेंट है।
ओके, तुम क्या करना चाहते हो? ये क्या करने की कोशिश है? आर्यन, तुम सुन पा रहे हो हमें?
या
हाँ, तो मतलब क्या करना चाहते हो? ये जो सॉइल तुमने ली है। हाँ,
या मैं इसमें 1 वीक बाद मैं इसमें स्लाइड पे डाल के इस पे कैमरे के जूम करके मैं देखना चाहूंगा कि इसमें रोटिफर आता है कि नहीं।
अच्छा, ओके। और ये हमें 3 बॉर्डर्स दिख रहे हैं, तो ये अलग अलग जगहों से तुमने लिया है क्या?
नहीं, ये मेरा नहीं है।
just showing for reference.
नो,
my tube
ये
lab.
मुकेश शो कर रहा है।
अच्छा, तुमने टेस्ट ट्यूब में किया था। ये तो इनका प्रेजेंटेशन है।
या मैंने आपको दिखाया।
जी।
हाँ, ओके। कैसा लगा आप लोगों को?
बहुत ही अच्छा है। मैं ये कह रहा था कि हम लोग सब उत्सुक है देखने के लिए कि तुम जब ये स्लाइड पे लेके या जैसे भी लेके तुम देखोगे, तो प्लीज ये फोटो जरूर शेयर करना हमें। हम सब देखना चाहते हैं।
या मैं आपको करूंगा।
हाँ, रोटिफर्स और उसके तुम्हें लगता है रोटिफर्स दिखेंगे और उसके अलावा भी कुछ दिखेगा तुम्हें?
अभी तो मैं नहीं कह सकता, लेकिन जब दिखेगा तो मैं आपको फोटोज शो करूंगा।
फोटोज तो ले पाओगे ना अच्छे, तो हम देखना चाहेंगे।
या
अच्छा आर्यन, मुझे आपसे कुछ कहना था। हेलो।
या
आप थोड़ा सा एक और बॉटल ले लो।
ओके।
एक और बॉटल ले लो और उसमें डिक्लोरिनेटेड वाटर ले लो। इसको ऐसे ही रहने दो। और इसमें से थोड़ा सा पानी इसमें से 2-3 ड्रॉप्स, जैसे अपने दूध का डालते हो ना 1-2 ड्रॉप्स, इसी तरह का इस पानी का ड्रॉप उसमें डाल देना।
ओके।
हाँ, क्योंकि आपने एक चम्मच का मिट्टी बहुत ज्यादा हो जाता है एक 250 ml वाटर के लिए।
ओ
हाँ, तो मुझे यही लगता है कि सेम एक्सपेरिमेंट आप रिपीट करोगे तो आपका रोटिफर आपको आंखों में देखने को मिल जाएगा सीधा।
ओके।
क्योंकि अभी आपका मिट्टी में ज्यादा मिट्टी हो गया ना पानी में,
हाँ
तो इसलिए आपको रोटिफर अगर होगा भी तो आपको नेकेड आई से बिल्कुल दिखेगा नहीं।
ओ
हाँ, तो ये मेरा सुझाव है। आप लेकिन आपका
या मैं
एक्सपेरिमेंट
कर लूंगा।
हाँ, कर लेना।
मैं ये भी करता हूँ और वो भी करता हूँ।
हाँ, दोनों करो, ठीक है।
ओके।
अच्छी बात है, अच्छी बात है। अरे हम लोग सब देखना चाहेंगे।
या
गुड वर्क, congrats. Okay. So we also saw this work. Aryan is very excited with this particular experiment that he wants to do. So we could have taken up a few more questions over here with Roti first, but then certainly we had a very long and engaging discussion, which we would really be looking forward that we have it on the group. Now, moving further, we would want the Moina group to do their presentation. And to do that, we again have Zara, Saida, Priyanka. So without wasting any further time, I ask them to upload their presentation.
Yes, I will give some short introduction. All right. Okay. Good evening, all. Good to see Aryan start with his home lab in Bihar, and it's a great inspiration for all. So we are going to present now the Moina model, and in this video, we will talk about the research we have done till now with Moina, and collaborate with all of them. Yeah, thank you. And I would also like to say that we did a goof up while making this video, that we forgot to add the further plan of work, so we can discuss that in our question answer session. Thank you.
Hi guys. How was
Hello
lockdown
everyone.
so far?
Hello everyone. Hi Jaswant. Hi Saida.
Hi.
How are you everyone?
Hello
everyone.
everyone. Hi. And say what's going on? Any news?
I got Moina from my college, and I
Oh,
don't know.
that's great.
And I don't know how to culture them.
Hey Ram,
Okay,
why
no
do you
worries.
worry when I am here?
Can anyone help me?
Yeah, why not? When I am here, why do you worry about anything? Let's see this video and you can get very well.
Yes, and it is very easy, and you can also do it at home. So yeah,
That's true.
yes.
Hi guys.
Hi everyone.
Hello.
Hi.
Hello.
Ram, why are you looking so sad?
Maybe I think I had made some mistakes in culturing the moinas.
What mistake you made, Ram? It's so easy to culture Moina.
Yeah, I know, but I had added few more drops of milk. I mean, I had added 4 drops of milk in the culture cup, but
Oh, what I did. I just added 4 drops of milk. I don't know what will happen next. Let's see. Hey, what is this red in this cup? Oh my god, this is Moina. But how could be this red? What happened to it? Did I did some mistake or what? How could it turn red? I have to talk to my collaborators about this.
4 drops of milk. Did the moina die?
yes. No, they didn't die. Actually, they turned red.
What could be that red color?
When I went to Munnar, is a high altitude region, my cheeks turned to red. But when I returned back, the color of cheek turned to normal.
Kashmir people are known for their red cheeks. Why is that so?
Yeah. And then what is red in human?
Yeah, it is obviously blood.
And blood contains hemoglobin.
So, do you think that moina contains hemoglobin?
Okay, let's find it out.
Hi
Hi
everyone.
everyone.
Hello.
So I'm stuck in yesterday's discussion.
What are you stuck in?
So how cells are coming to know that it's time to produce more hemoglobin?
Yeah, and it is such a tiny organism, and then so it changes its color.
Yes, and interestingly, guys, I found that the 2019 Nobel Prize had gone to the studies of this particular mechanism.
What exactly happened?
Yeah, I'll just show my screen so we can have an idea of that.
Yeah, it looks very complicated.
Oh, it
What
is
is
not much complicated if we try to understand.
what is normoxia here?
So we can break down the words, Ram. So norm means normal and oxia means oxygen. So normoxia means normal oxygen condition. And I
And
guess you must have figured out about hypoxia.
yeah, I suppose it is low oxygen level, but I don't know.
Yes, hypoxia means low oxygen level. Correct.
So then what is HIF here?
So yeah, HIF means hypoxia inducible factor. It is a protein.
So does HIF get activated in hypoxia?
Yes, exactly. That what its name suggests. Hypoxia inducible factors get activated in hypoxia only.
But does that mean that HIF protein does not bind to the gene normoxia?
Yes, as we can see in the graphic, the HIF1 alpha, the protein is getting degraded in normoxia.
What is the role of VHL here?
So VHL is acting as a tagging protein which is being binded to HIF, after which the degradation of HIF happens. So because of VHL,
So,
HIF is getting degraded.
so Dristan, what is the HIF is doing in hypoxia condition here?
Yes, so that's what the question is. So HIF is going into the nucleus and it is binding to our hemoglobin genes. The genes earlier which were inactive now will be now they will get active when HIF is binding to them.
The diagram indeed look very complicated, but yet it is very much simplified.
Yes, this is what the Nobel Prize and Moina have the relation.
When I added 1 drop of milk, the moina was colorless, but when I added 4 drops of milk, it became colored.
So what if we add that colored moina in 1 drop of milk culture? So will this colored moina will remain colored or it will turn colorless?
Guys, we know that many people suffer from diseases wherein they have breathing problems due to the lack of oxygen carrying protein called hemoglobin. So I was wondering that is there any way to study the continuous production of hemoglobin through Moina?
Yes, guys, and one of my uncle who is suffering from epilepsy, and he is taking valproic acid, and I have searched that it is HDAC inhibitor.
Does valproic acid inhibit histone deacetylases?
If so, can we use this valproic acid to keep moina colored in 1 drop culture?
Hi guys.
Hello everyone.
Hi. Hey guys, look what I found in my moina culture. I'll just start presenting my screen.
It seems like egg.
Guys, do moina lay eggs?
No, I don't think so that Moina lay eggs because I have seen them give birth to live progenies. I'll just show my screen. Here you go.
Oh wow, here we can see that the moina is giving birth to new progenies.
Guess what, guys? While you're a little busy looking into the moina giving birth to young ones, I searched the internet and found that under stress conditions, moina does lay eggs when it's subjected to hypoxia, that the condition of oxygen is less, or when there's overcrowding in the culture, or even when it's exposed to toxic environmental conditions, it does lay eggs.
So we can induce hypoxia as we are already creating hypoxic conditions for moina to get colored condition, isn't it?
Yes.
Yeah, we can also use moina as an indicator. Like moina give lay eggs only in unfavorable condition. So we can if moina is giving egg, then we can indicate that the moina is not the condition where the moina is not good for moina.
I was thinking that, as Avipsa said, that Moina lays egg in stress condition, and we already are doing that, right? So why don't we go ahead and design an experiment for that?
Yes, that's a good idea.
Yeah.
Okay, so yes, I really liked the way you in this particular video, through a conference call, you were trying to, build up your knowledge, build up your understanding of what you're doing, and trying to understand the concepts. But one of the members in your conference call, Seda, I don't think through those conference calls all her questions have got answered, because she's constantly asking lot many questions in the chat over here. So Seda, could you just unmute and ask the questions whatever that you were typing over here?
Yes. So firstly, we just started with Ram getting the moina, and we were explaining how do we culture it. So can anybody in the group? I know I'm a group member itself. Sorry for asking questions, but that's what we do in Cube. So can anyone tell me how do we identify moina? How do we say that this organism is a particular moina? Yeah, how can we identify moina? Can anybody tell me? Justant, Rachel, Man.
Yes.
Okay. Can we go to the other slide where the photo of Moina is shown? Okay, the other one, the one under microscope. Yeah. Okay.
So here we can see some features like we most of the time we cannot differentiate between Daphnia and Moina, but we can see a large antennule. By seeing it, we can identify that it's Moina. So here we can see the 3 different species of Moina, and all of these are having antennules.
Rachel, can you point out, because people have joined from different parts of the country and they might not
Yes.
know about moina. So can you specify the moina, specify the antennule that you're talking about with your pointer?
Yes, so below the eye there is antennule. So in Moina micrura, also in Moina brachiata, and also we can see in Moina macrocopa. So this is how we differentiate between Daphnia and Moina. So yes.
But I can't see any Daphnia's photo. Sorry, yeah, Daphnia's photo. They are all Moina, right?
We have
Hello.
another slide. Saida, can
Okay.
you go there? No, the other one, which has the comparison between. Yeah.
So
Yeah.
in this, yes. So in this slide, we can see that uh, Moina is having antenule and Daphnia is not having any antenule. Uh, so there is a beak-like structure or the pointed structure. There is no antenule present in Daphnia.
Okay, one of the question, like during the discussion, we came up with what is red in humans, and it could be hemoglobin. Is Moina actually producing hemoglobin? So I just wanted to ask: is there no hemoglobin in colorless Moina because it turned red, right? It turned colored later in hypoxic condition. So in normoxic condition, is there no hemoglobin produced at all? Hello.
No, we can't say that no hemoglobin is produced because hemoglobin is the protein which carries oxygen. So if we say that no hemoglobin is produced, then no oxygen will be carried, so the organism won't be alive. So hemoglobin is being produced, but it is being produced in normal amounts, so that it is the organism itself is appearing to be colorless. It is being produced, but in normal amounts.
So we have
So in,
yes, Shashi.
so in normal amounts, is the hemoglobin not red in color? What do you mean by colorless? Why is it colorless?
So if we
What do you
compare,
What
okay,
do you mean by normal amounts? Like, I didn't get you.
if we compare with the colored moina, so if we compare a colored moina and a colorless moina, we'll see that the colored one is appearing to be, I mean, by the color only we can say that more hemoglobin is present in that organism. And if we compare that to this colored moina, colorless moina, we see that comparatively less hemoglobin is present. So we have not estimated the amount of hemoglobin present, but yeah, we can say in qualitative terms.
So you are saying that even if the hemoglobin is present in very low amounts, it won't be red, because the Moina with like the normal in the normal conditions, it is not red. It is not seen red. So I don't know. Like, what do you think that
So
that is
for me,
after the red?
yeah, I know I asked the question and I am only answering, but
Okay. Okay.
yeah, it sounds absurd, but now it got clicked to me that even humans, um,
Yeah.
when we in the this thing also in the video call also we kind of highlighted that humans in normally humans don't have red cheeks or red ears, but when we go to a higher altitude we turn red. Does it mean that we have no hemoglobin in the normal condition? We still have blood, right? If you and we still have hemoglobin, but it is in an amount that the body is not seen red, because not it's like an amount where the body is not turning red because hemoglobin is
So why
comparatively.
do you think? Okay. Okay. Yeah, I get you now.
So my question is that the hemoglobin is red. In both the conditions, the hemoglobin as a protein is certainly red. So what is it about the higher concentrations that we are able to see the organism being red, or even with humans, the cheeks being turning red? Is there some physics to it, or what? So that the same hemoglobin, though it's red in color, when it's in some lesser concentration, the organism macroscopically we do not see being red. So is there some physics in this that explains this?
So, if I was to say it, you can say when what is hemoglobin is actually hemoglobin is an oxygen carrier. So when there is normal amount, ample amount of oxygen, we don't need more hemoglobin. And when there is less amount of oxygen, more carriers are produced, more hemoglobins are produced, so that the less amount of oxygen is somewhere carried like passed on in the body. So that's why there is an increase in hemoglobin level at low oxygen. Uh, yeah, that's what I think.
Okay. So, is this hemoglobin the same as the that we find in humans?
I have
Same as
an
in?
answer to this.
Okay, go ahead. Go ahead.
So if you have seen when people run also, so some people get red or a bit pinkish in color. So it's the same when we see the bottle in the bottle, the colored moinas when they are in red in color. So when we are running, it's because of we are consuming more oxygen. We want more oxygen. So it's the same with moina as we are comparing it.
So here we
And
are not taking yes. Okay, so what I was saying is here we are not taking Moina to a higher altitude region for making the level of oxygen less, but instead, like in the video also you must have seen, it was just by increasing more milk, as in because there is, and to clarify, milk is not uh, Moina doesn't feed on milk. Moina feeds on bacteria. So when there is more milk, more bacteria will be produced. More bacteria will need more oxygen and will take up more oxygen, because of which there would be uh, hypoxic condition. Yeah, so that's how point is turning. It's not only about high altitude region, it's just the thing about low oxygen level. Now now you would one of the question would be like, how do you measure oxygen, how do you say the oxygen is less? So in our group we have mentioned that we have not experimentally done it, but yeah, our seniors and our former cubists have actually tried doing biological oxygen demand, dissolved oxygen. So they found out that there is the oxygen, the level of oxygen does go down when you put more drops of milk.
Okay. So, with the later part where you talked about how the genetic mechanism part, where you where through a conference call you were trying to discuss how what is the mechanism. Could you explain how oxygen, plays a role in deciding? So, if you could go to that slide.
Yes, yes.
Yeah, this part. So those oxygen molecules coming in, and how is that? Could you just shed some light on this part?
Okay, okay. So this, just a minute. Yeah, so we here, so first we'll talk about this protein, which is called HIF. So HIF is a protein which has the function. So HIF means hypoxia inducible factor. That means it gets activated only when hypoxia is there. But when there is no hypoxia means less oxygen condition. But when there is no less oxygen condition, there
So
is
you could first
normal.
explain what happens when there is hypoxia. What is that HF doing when there is?
All right. Okay. Okay, so as we can see in hypoxia, the hypoxia inducible factor is going inside the nucleus from the cytoplasm, and it is getting bound to the DNA. So here we'll talk about the hemoglobin genes. So it is binding to the uh, genes of hemoglobin. In our case, in Moina's case, it will bind to the hemoglobin genes, and it will get, so the genes will get the signal to start producing the hemoglobin protein. Now we can see there's something written HRE. So HRE are nothing but DNA segments. HRE means hypoxia response elements. So when HIF is coming and binding to these HREs, that are hypoxia response elements, the genes start producing the mRNA. I mean, DNA will get converted into mRNA, and mRNA will be translated into protein, and we get the hemoglobin protein.
So
But
Drishta,
now,
over
yeah,
here, in normoxia conditions, as well, hemoglobin is found in the blood. So hemoglobin is being produced. So
yes.
is there anything else other than HIF which is allowing that normal level of production of hemoglobin? So how is the hemoglobin otherwise produced in normoxia?
So hemoglobin has in Moina, hemoglobin has, so there are 4 genes of hemoglobin in Moina, and in the normoxia condition or normal oxygen condition, only 1 gene is producing hemoglobin, and the rest 3 genes are not producing hemoglobin. So that is why we get the normal hemoglobin through that single gene.
So all these 4 genes of hemoglobin produce the same hemoglobin molecule, or these are different kinds of hemoglobin molecules?
No, these are the same hemoglobin molecule.
Okay. Now you can tell about the normoxia condition.
Yes. Yeah. Now we can see that in normoxia, where there is normal oxygen condition in the cell. So this is a whole cell, and when there will be oxygen in the cell, we can see O2 molecules here. The O2 molecules. So one of the oxygen molecule will bind with an will form a bond with the hydrogen, and it will create a hydroxyl molecule, that is OH, and that OH is binding to this HIF protein, the same HIF protein which was in hypoxia binding to the nucleus genes. And now when hydroxyl is binding to the HIF, further the VHL, it is a protein, again VHL is a protein, VHL will bind to this hypoxia response, hypoxia inducible factor HIF, and then VHL will take it for the degradation, that is proteasomal degradation.
So do you mean to say that the VHL does not bind unless and until oxygen is there? Does not bind to HIF?
Yes.
Okay. So coming back to our question, as in our further objective, which is not quite mentioned in the video very well. So we hypothesize that again the 4 genes of hemoglobin are there. There is some histone remodeling happening, as in, so we believe that this is our hypothesis, that one gene is always continuing to produce hemoglobin, and the 3 genes are bound with the histone. Now we of course know histone and DNA bind together to form. So
Saida,
yes.
we can actually show the reference which says that these genes are the next reference. Again, one more slide. Yeah, here we can. So can you turn on the? So this part explains that only 1 gene. So you can continue with this slide. The highlighted part explains that.
Yes. Yeah, only 1 gene is producing hemoglobin, and the rest of 3 are bound with the histone. And of course, the histone is positively charged and DNA is negatively charged, so that's why they are bind together to compress the DNA and fit inside the cell. So we expect that in hypoxic condition, what happens is this. There is something called as histone acetylase. So that will kind of attach.
What is compression? What does compression has to do with expression? I don't know, DNA being compressed. So what?
So as in that gene. So what I meant to say was that gene, the rest of the hemoglobin gene are bound with the histone. Okay. So the gene is not expressing, so then more protein is not being made, more protein is not produced, so there's just normal hemoglobin that is being produced every now and then. But when there is hypoxic condition, we expect we hypothesize. I would say that we hypothesize that this histone acetylase will attach acetyl group on histone, making it negative. So because DNA is negative, again histone has become negative. They will repel each other, and because of which the DNA will be free, and this will allow RNA polymerase to read the DNA segment and make RNA and then further make protein. That's how protein is made. And one more part, that when moina is again put into 1 drop of milk, it will turn colorless. Like what we said, when Arunima came back from Monar, her cheeks again become colorless. So how is that happening? Is we think that there would be some histone deacetylase that will remove the acetyl group, making again histone a positively charged protein. Again, histone and DNA will bind together, and this gene expression would stop, because of which there would be no more protein made, more hemoglobin protein made. So this is our hypothesis. So we are actually, as we mentioned in the video as well, that we'll be trying to use valproic acid, which is an HDAC inhibitor. So if we use HDAC inhibitor, what we want to do is we want to keep in 1 drop culture, we want to keep HDAC inhibitor, like put HDAC inhibitor, valproic acid, and we expect that the moina will remain red. So by not cutting the moina, by not actually killing the moina, we can find out what is happening on the molecular level. That's what our plan is.
Okay,
Yes.
great.
Yes.
So could we now, yeah, Grisha.
Yes. So in the video, Anjuel was talking about anti-epileptic drug called valproic acid. So yeah, we are using valproic acid in moina also for experimenting with moina, and this is the reference that tells about valproic acid inhibiting the deacetylation in histone.
Okay. Okay. So could we, as a last part, could we discuss the concept map to draw some connections to your curriculum?
Yeah, one part before continuing the concept map, I would like to add that we are just not asking about what is happening on a molecular level in hypoxic condition. There's also one more question being asked, as discussed in the video, that moina switches its mode of reproduction in stress condition. And this stress condition could also, hypoxic condition could also act as a stress condition. So yeah, it's a cool thing. Yeah, we can continue with
Good.
concept map.
Yes.
So if someone could briefly explain this, very briefly explain this.
Yes, we have various subjects of our related with moina. So we can first come to a research methodology, because in our curriculum this year we have this subject wherein we study the development of experimental design, then having controls, test, and the sampling methods in that, and then we go to the most important part of this lockdown we learned is about the home lab microscopy, through which we can actually magnify the see a magnified view that organism through our phones by developing some joints as we and then we see that as we talk about the bacteria and law of oxygen, we also talk about the biochemistry of that. How is the bacteria lowering the oxygen? I mean, what is it consuming, and what is happening inside the dissolved inside the water which is there? So we can do experiments such as dissolved oxygen experiment, or actually calculate the biological oxygen demand through experimentation. And the most important is 2019 is actually related with moina, and we discussed it right now. And as always, genetics also forms a very important part of our curriculum. So we also related it with Moina. And central dogma has been always been with us in every model organism. We discussed it in all of the organisms. And the most striking, we can say, a topic of frontier research is frontier research related with Moina is epigenetics. So this is a new field of science, we can say, wherein we actually study the phenotypical changes in the organism without actually altering its DNA sequence. So we saw that as we kept Moina in normal condition and then in hypoxic condition or low oxygen condition, it changed its color. And the surprising thing is when we keep that colored Moina back in the normal condition, it will turn back to colorless. So we can see that without actually studying altering the DNA sequence or the DNA sequence, we are studying the characteristics, that why is it happening and like that. Yes.
Okay.
Yeah.
Okay, great. So thanks a lot, the entire Moina group for your presentation, and later on with the discussion that we had, specifically touching upon this epigenetics part that Dristant just mentioned. We talked about acetylation, this chromosomal chromatin modification and all. So very interesting stuff, and we look forward to a few more such questions being taken up on the group with regards to this. So thanks a lot. Thanks again for drawing out so many connections. There are a lot that we get to see so many different topics being touched upon by this single, by working on this simple single organism. So thanks a lot. And so now we move on to our next presentation, and that would be the mosquito group presenting their work. They will be talking about the awareness that we need to have with regards to mosquito and the diseases that they cause. So over to who do we have over here, to Hina ma'am and Rahil. Over to you for the presentation on mosquitoes.
Asutosh?
Yes, DP. Yes, DP.
Asutosh, am I audible?
Yes, you're audible. Could you just hold on with your presentation? Yes, DP, go ahead.
Yeah, so sorry, I think I was following on the YouTube. We will be launching chatShaala.in right now. GN will briefly tell us about it, and we'll see a short video, and then we will move to mosquito thing.
Okay. So with the mosquito group, can you just hold on for a while? We will have this particular session by Nagarjuna sir, and then we can proceed.
Yeah. So
yes, DP, you want
yes,
to say?
GN, I think you can go ahead, and I will take over the presenter, and we will show the video after you talk, or whichever way.
Yeah, you can load the thing. You can hear me, right?
Yes.
Okay, so what I'm suggesting is you load the video. I'll just say a few words.
Okay.
Basically, inspired by the chatShaala that are happening relentlessly for almost 7 months now, and then we thought that it's a very good idea to bring all the discussions that you guys are having in WhatsApp and emails and in various other groups, and so why not have a threaded discussion of all the conversations, so that it will be easy for us to archive the quality discussions that are happening throughout, both offline and online conversations. So keeping that in mind, at Homi Bhabha Center, the Knowledge Lab, our colleagues as well as some student interns. Just like you guys are working on biology, there are some student interns who have been working at Knowledge Lab from BITS Pilani. So, and then some of our own software engineers who are working with us have developed a special chatShaala app for all of you. And so we encourage all of you to use this application so that all our conversations are available, and it is developed in such a way that while you are using chatShaala.in, the messages will be automatically archived on metastudio.org directly. And of course, people who use metastudio.org can also be available through chatShaala.in as well. So it's a kind of single archive, single database shared by two different applications. And it is a 100% mobile friendly application, so you can actually load chatShaala.in, and it has a very similar interface like WhatsApp and Telegram. So that's all I want to say now. So we want all of your encouragement to start using chatShaala.in so that we will be able to archive all your conversations. You can upload pictures and have discussions, but based on each topic. So that's what I think DP's video will be able to convey that. I think the video is been created by Ravi. Is Ravi around?
Yes, he's around.
Yes, yeah.
Yeah. So thanks to Ravi who made this video. So it's again a big collaboration between our lab people, BITS Pilani students, and so many others who contributed. Go ahead.
Yes.
Yeah, just to say, I've been just reading some other question. One of the question asked was, uh, if they already have an account on MetaStudio, you can use the same account and the same passwords. Yeah, as I said, both of them actually use the same database, so you don't have to create a new account. But if you don't have an account on MetaStudio, you can create a new account by using your email. Yeah, I mean, if there are no more questions on this, we can go ahead with the next presentation. Maybe tomorrow also we'll show this once again, because we don't have all the participants today at this time. So we may do it tomorrow again after the first session,
Yes.
so that other people will also get to know about it.
And even in the last part, like when we are doing microscopy workshop today at the end, we can have quick hands-on activity for all those who already are active on MetaStudio. So I know some of you who are active, so you can try it out right then and there when we show certain things. Definitely. Yeah. Over to Ashutosh.
Yeah. So I would simply say that the Mosquito group can continue with their presentation now.
Long-term objective: to spread mosquito awareness and the diseases spread by them. Now the question arises that why is a deadly creepy mosquito a model organism? But before telling you that, we will tell you about types of mosquitoes we'll work upon.
Aedes albopictus mosquito can be identified by the straight line-like structure on its upper head region. It is a vector of both chikungunya and dengue virus. It is also called the Asian tiger mosquito. Aedes aegypti mosquito can be identified by the lyre-like structure on its upper head region. It is the main vector of dengue and yellow fever. It is also called the yellow fever mosquito.
It's a dark brownish mosquito. It sits on an angle of 45 degrees with its lower part of body upwards. It is the vector of malaria and is also known as marsh mosquitoes.
Now let us go back to the main question, that is, why is a deadly creepy mosquito our model organism? Mosquitoes are easy to work on. We can find them anywhere. We can find them in any water accumulated place and can identify them easily. Mosquitoes can be found in tropical and subtropical regions of the world. Here is the data on the disease transmission of Aedes aegypti as of year 2019.
As compared to other mosquitoes, Aedes aegypti is simple to breed and has therefore been the subject of a wide range of research investigation.
From the data collection of our home lab in 2019, number of Aedes mosquito were more than non-Aedes mosquitoes. In October 2019, we found 25 Aedes and 17 non-Aedes mosquitoes. In November 2019, we found 34 Aedes and 21 non-Aedes mosquitoes. In December 2019, we found 18 Aedes to 11 non-Aedes mosquitoes.
General mosquito breeding places in our home labs.
Generally, while watering plants, the water gets accumulated in the planter plates. The water is sufficient enough for the mosquitoes to lay eggs and breed. Here also we can see water in planter plates. Sometimes water also gets accumulated in buckets used for watering the plants, and on various occasions we have seen larvae and mosquitoes there. Also, in planting pots, water gets accumulated. We can also see an Aedes albopictus mosquito on the wall of the plant pot.
Two mosquito friends, Joe and Mill, talk about their current situation.
Hi Joe.
Hi Lily.
This pandemic COVID-19 has taken away all the importance which we mosquitoes were getting from spreading chikungunya, dengue and malaria fever.
Yes, very true. You are right.
Joe and Mill looks for breeding places. Joe and Mill talk about their general problem faced by them while spreading dengue and other viruses.
This bacteria, Wolbachia, stays in my stomach and makes my friend dengue virus inactive.
Yes, Millie, you are right. This is checking our population.
Joe and Mill talk about the prevention taken by humans.
Humans are using biocontrol method to check our population.
What is biocontrol method?
These humans add gambusia fish to water bodies, which kill our beloved larvae.
In 1902, Ronald Ross won the Nobel Prize for the discovery in 1890s on malaria-causing parasite and role of Anopheles. Alphonse Leveron got the Nobel Prize in 1907 for his finding that malarial parasite is present in human blood. Paul Herman got the Nobel Prize in 1948 for his discovery on the role of DDT in control of malaria. UU2 got the Nobel Prize in 2015 for the extraction of substance artemisinin which inhibited malarial parasite.
Joe,
Hello, Millie.
this pandemic COVID-19 has... Hi Joe.
Hello, Millie.
This pandemic COVID-19 has taken away all the importance which we mosquitoes were getting from spreading chikungunya, dengue virus.
God.
Okay. Yeah, ma'am, you have something to say? A very nice video.
Yeah.
I like the video.
Thank you, Ashutosh. Actually, we've been working on mosquitoes since the last 3 years, and it all began with just a simple survey to try to identify the mosquito breeding places in 2016. So, like, it is slightly an evolution for the children to learn to find out the difference between Aedes and non-Aedes and the identifiable features. And so this work is basically going to go on with the types of viruses. I think the long-term objective they are going to find out now is along with mosquito prevention and control awareness, which is very important in our city, where children have been working in the local vicinity where they reside. During lockdown, we couldn't, move much, but yes, they were working in their vicinity, in their residential areas. And now we are going to work upon different types of viruses they are carrying, and the research work by the students will be done there. So, any questions, like if the audience has, you may ask.
So even before we take up the questions, ma'am,
Mm.
we saw that in the video they had this very very nice, funny goof ups while shooting the video.
Yeah.
But then what I wanted to ask is the what we really look forward to is the goof ups that they had during their experiment. So any such goof ups that the presenters would like to share, and we could talk about it.
During the experiment, during the survey and all.
Yes, ma'am.
Yeah, yeah. I think Vani would definitely share how Rahil can also share how. Yes, please.
I have one goof up while clicking pictures. Once, when I was clicking pictures from a pot, the whole pot turned over and the pot also broke. And because of that, my neighbor was totally mad at me. That why did you do that? And all these things happened.
Okay, okay. So we could take up a few questions from the by the participants over here. So Kunal is asking, can we have control measures and reduce mosquito breeding places? How?
Yeah, we can control mosquito we breeding places by if we see larvae in some water, stagnant water or any accumulated water, we can add drops of oil in it. It will kill the larvae instantly. And some other control measures for the adult mosquito, the flies. We can use sprays and many other things. And
If
if
you
you
okay, okay.
Even we can add gambusia fish to water bodies if we find it near us, because they kill the mosquito larvae.
So Sarita ma'am wants to ask, why does oil kill the larvae? How does that happen?
when we add oil in the water, the oxygen no, when we add oil in the water, the larvae are unable to breathe because of the thick layer of the oil
Thick layer of the oil on their body or where?
on the water surface.
So are there any so in terms of public awareness, do you have any plans ahead of how you would increase the awareness in the public with regards to the mosquitoes?
uh, we generally tell our neighbors, especially in our home lab area, that when you water plants, so control the amount of water they are giving to the plants, because if there is more water and the water get accumulated in the planter pots, and generally I find mosquitoes there only, and even in some empty buckets out there.
Okay, okay. So in case of the mosquitoes, they take up atmospheric oxygen. The larvae that are growing beneath the water surface, they require the atmospheric oxygen. That is what you mean to say when you talked about the oil as a mechanism of killing the larvae.
Yeah, I mean that only because the larvae come up to the water surface to take air from their siphons.
Okay, so they do not take in dissolved oxygen. They need the atmospheric oxygen. That is what you mean to
Little
say.
bit take I think so, from the water, little bit.
Okay, okay.
Yeah.
Just
Now
connecting
I
with
had a
Moina.
question. Yes,
Sorry.
Saida.
Yeah. As in QB, kind of collaborate and you know connect two or three organism and ask a similar question. So I was just wondering, this oil that we talked about right now, will this oil, because if the mosquito larvae and stuff also need oxygen, will this also create some hypoxic condition, and because of it the larvae will die later because there would be no oxygen as such because of the oil layer?
Yes, yes, definitely. I had another question to the team. I'm behaving as a audience. I want to ask that in Homi Baba lab, you people had visited the Pagalopus wells outside, and from there in the well we had found larvae. Can you tell why did we find mosquito larvae in the well, in the Pagalopus well? I think Vani, Rahil, all of you had done that. Can you just tell why?
Yeah, yeah. We
We found
found
stagnant
lar
water over there, and the atmospheric was suitable for the mosquito to lay the eggs as well. And we found larvae because since the water was stagnant, so we could get algae, bacteria, and protozoans over there also, because they serve as a food for the larvae.
So that means an insect lays egg in water. Okay. So why does it lay egg in the stagnant water? What do you think is the future? The eggs will hatch into what? So can you please explain how come Pagalopus well is a suitable site for breeding of mosquito? A very small little well of maybe 6 centimeter or so. Can you just please explain?
there are bacterias and algae in the water, so that the larvae can eat them. That's why we find in them in Pagalopus wells.
Okay.
So
So
I visited Homi Baba lab during monsoon season when there was rain, and because of which the water got collected in the Patalapus well as well. Um, that could be one of the reasons. So
mhm.
is it really about Pagalapus well, or is it really about any kind of stagnant water, anywhere the water is collected? Anywhere the
The
water is
mosquitoes,
collected,
yeah, anywhere.
anywhere.
But probably the Pagalopus well acts as a good source of nutrition for them. Can we say that?
Yes, we can say.
So Vani, how can you explain the food chain? Like what kind of food is available for the larvae to feed in the Pagalopus well, that small little well? And you people had made videos of the larvae. So this question, what kind of food do you think the mosquito larvae must be feeding there?
Algae, protozoans, bacteria, because if the water is stagnant over there, then we might find some organic matter also on which the larvae feed.
Okay. So what is your like? What do you intend to work further on? Like this model organism mosquito. What is your future planning? What do you plan to work upon? The different types of viruses which you were discussing, that is it an RNA virus or a DNA virus? So what kind of virus is dengue?
For the YouTube question answer, uh, did you culture mosquito or trapped only for identification? No, I didn't culture mosquito. I used to go outside our home lab and find stagnant water because many times I have seen mosquitoes and larvae there. That's why I used to click pictures there. We don't breed mosquitoes here. Otherwise, the municipal corporation will beat us up.
Okay. So when you used to observe these places where you had stagnant water and you observe larvae, you found Aedes and Culex. How do you identify, differentiate between the two larvae, Aedes and Culex?
Culex siphon, I really don't identify the larvae sometimes, but the larvae, as far as I remember, I find only Aedes mosquitoes over there. So hopefully there are Aedes larvae only there, because I have never seen any Culex or Anopheles mosquito there.
Okay, so could there be any competition between the Culex and the Aedes mosquitoes, or is it like the Aedes larvae have completely outcompeted them?
Uh, yeah, as per the data, also we have seen during these months, Aedes mosquitoes are more because of the rains around the our area.
Okay, okay.
Non-Aedes mosquitoes, non-Aedes mosquitoes are also there, but in a very less amount. Like for every uh, two Aedes, we find one non-Aedes mosquito. It's like that. And during the evenings, they sometimes come inside our home and we kill them.
But then your data table showed both these Aedes and Anopheles. If we go through your presentation,
Yeah. The data showed Aedes and non-Aedes of previous year October, November, and December, 3 months data. There is a slide also. Can you please show it? Yeah, from the data we can see during October there were 25 ADs which I was able to capture photos of, and 17 non-ADs. Here also ADs are more in numbers, and in November there are 34 ADs and 21 non-ADs. And in December there are 18 and 11. The number decreased because of the cold temperature of both the mosquitoes, and the total mosquitoes were there 77 and 49, 77 ADs and 49 non-ADs in whole 3 months.
so
So
what do you mean by
comparatively,
non-Aedes? As in, we are not able to understand. Non-Aedes means
like Aedes mosquito have bands, white bands, and white shapes on their body, whereas non-Aedes have no white bands. They are dark brownish in colors generally. Like
non-Aedes is a species. You mean to say?
Non-Aedes is Culex and Anopheles. Here
So
I mean
both.
to say that.
So okay. So both have the same feature. You mean to say when you say non-Aedes, Culex and
Not same features but different. Their identification parts are also identification signs are also different.
okay. Okay, so this particular data is from where? This the table that you're showing.
Our home lab,
Home
our
lab.
home lab in the
Yeah,
garden
you
area
were
and
mentioning
the
the way.
water accumulated area.
Okay, okay, okay. Great, great. So nice work. To your curriculum.
One.
Yes, ma'am. So, life cycle of Aedes mosquito, if I discuss, the eggs are laid by the female mosquito, and approx 200 to 300 eggs are laid per batch, and then the larvae are formed, and within a time span of 7 days, it changes into the pupa, and after 5 days, an adult mosquito is formed. And then we have the pollination mosquitoes also serve as
a source of pollination from transferring the pollen grains from the anther to the stigma. And the diseases caused by the mosquitoes are yellow fever, major vector. It is a Aedes usually causes dengue. It is the main vector. It also spreads malaria and Zika virus also. And
Aedes spreads yellow fever, dengue, and Zika, whereas
malaria
Aedes
is spread by Anopheles.
malaria. Yeah,
By
and they
Anopheles,
all
yeah.
are the vectors.
Yeah, they all are the vectors. And then the habitat, uh, um, the environmental interaction. Different species of mosquitoes require different temperature and different types of water to breed. Mosquitoes usually live in dark, shady, and damp areas. And then mosquitoes belong to the second trophic level, and they are the primary consumers. And if we eradicate the mosquitoes from the food chain, it won't affect much, because since mosquitoes are the because they usually are a source of food for the birds, lizards, and the fishes.
So if they are the source of food, then that would affect, right? If mosquitoes are not there.
Yeah, that would affect, but not that much, but not to a very great extent.
How are you sure? How are you sure? You could go ask the fishes. Maybe they would tell you a different story.
No, sir, fishes can also feed on other things, not only mosquito larvae, but they can also feed on other things like.
But did you ask them what they prefer for dinner?
Uh, Not like that, but we'll say.
So there are a few more connections I get to see. This frequency statistics. I'm not really understanding this part. And
Actually, I'll just help out for that. The frequency and statistics is when they were collecting the mosquitoes. That is how the population dynamics part is. This is a map which tells how mosquito is related to our curriculum. So it is related because we are talking about disease, we are talking about pathogenicity, we are talking about our immune system. If you have good immunity, the virus will not affect. And then here the statistics and the frequency is when we are counting the number of, when they are talking of Aedes and non-Aedes, it's basically they are trying to, the first identifiable point was that let's check out for the striped mosquitoes, because that time it was the epidemic, and still the hospital beds are often full with dengue patients in the city. So we would always, the survey would always say that check out for the white striped band mosquito, and if you find them, just kill them. So that kind of a slogan was created, and a campaign was created in 6 schools. So that is why they are saying Aedes and non-Aedes. And this map itself shows how the evolution is linked to mosquito. We can talk about the changes, different types of viruses it has started carrying, and how the ecology. Uh, Actually, the children started explaining the detail, but it's how our subject is related to mosquito. So we can connect this model organism to our curriculum, and there are various places where we can connect to a mosquito as a model organism, and its application in subject also, in context also.
so that is interesting. Okay, nice.
So, Hina ma'am, I have one more doubt. Did these mosquito bite the dead human bodies and take blood from that? Yeah, dead
This is
bodies.
very interesting because when we talk of certain fossils of mosquito, that day I was discussing about fossilization in the class, and there was a story which I just hinted the children that sometimes we find fossils of mosquito which have blood in it, and it has been, in the ice we have found, and there were some facts the children were telling me. So this is what you're saying, that if they suck the blood and they die, we may get the DNA of maybe 400-year-old animal, or maybe the dinosaur mapping which was done. So it helps in study of genetics, it helps in study of evolution, it gives us a lot of evidences also, the fossils of mosquito. That is what you asked? Yeah.
Yes, yes.
And even there is a lot of mutation occurring, like the kind of viruses they have started, carrying, even their vector prognosis is changing. Like now I have given the children a new point of study, like let's find out the different types of viruses these mosquitoes are carrying and causing infection. Let's go into the detail, because coronavirus is itself an RNA virus, and why don't we compare the two? How it could have been related? Is there a is there a similarity? Is there a dissimilarity? So that is the future. Now this year they are going to work upon the different types of viruses the mosquito carries.
Okay. We would really love if we could have a very nice discussion on this, and even before you go about designing an experiment or something. If you could have a discussion, because it's
Yes,
something that's
Ashutosh,
very
because of
relevant.
lockdown, we couldn't bring a lot of experimental evidences to our movie, because children were not unable to connect to the lab and do a lot. But yes, we have collected a lot of genuine data which the children have done. All this photograph is from mobile camera. So the most important thing, except one or two Anopheles pictures, non-ADs, we have taken from the this thing. Google. Otherwise, all the pictures have been taken from the mobile camera. That is what we are trying to motivate the children for. We don't need big DSLRs and all. This is from the normal mobile camera that the pictures have been taken, and all Aedes mosquitoes. Because we are concentrating on the awareness of Aedes, telling people that see where they are breeding, and this is very dangerous. The amount of platelets which were needed in the city was, there were newspaper cutouts, and there were alarming messages and fumigations being done whenever we are posting messages to the municipal. I mean, it is actually we have worked upon the prevention and control at the school level. We have brought about a lot of awareness, because this school is in a very congested area. It's a pure residential locality. So
Okay.
catching mosquito.
So this could be connected with the whatever that we read in the news, because even now, as we get to see people saying over here that the dengue cases are increasing in Delhi and these areas. So the dengue is caused by Aedes.
Yeah, it is
Dengue is caused
carried by
by
Aedes. Yeah, it is spread by
Aedes.
Aedes. Right.
Okay,
Yeah,
okay,
it's a virus which is spread by Aedes.
okay, okay. So nice, interesting. So so we would really be looking forward to the work that you plan to do ahead, and even whether you do work in the home labs or not, the participation in these chat shalas, where even we get to see that in this concept where there are so many different topics that it gets connected to. So even if we continue with our discussions in the chat shalas, there is so much to learn with the mosquitoes. So good, nice presentation all, and we look forward to your work.
Thank you.
So yes, thanks a lot, everyone. So now we are left with this very last session in for today's day, and that would be on microscopy. So DP would be talking about it. Would brief us on what is the session all about. Over to you, DP.
Yeah. Thanks, Ashutosh, and hello everybody. Back again. So we have been discussing quite a lot of things today, from butterflies, cardamines, rotifers, moina, mosquito, and we briefly also saw the chatShaala.in app in between. So what we will do now is, because it's 9:15 now, we will be splitting the activities into two parts, and one part will have a kind of follow-up thing for you all till we join back. Yeah, so we'll just wait to get a confirmation from the stream as well.
The stream is continuing.
Yeah, stream is continuing, but they need to join. Yeah. Is Ravi here? Yes.
Yes, Giri, I was just checking with Surendra if sound is coming on YouTube or not. Can someone quickly check if I will also.
Yeah, I confirm that stream is working.
Okay. Okay, so what we are going to do is we are going to quickly, in the first part, gather some ideas that you already have, and we will be we'll be having a series of interactions in the coming weeks around microscopy itself. And the idea behind this is that okay, so Ravi, do you want to share your screen?
Yeah.
Yeah. So as Ravi shares his screen, so how many of you have tried to see some other thing on a microscope till now?
I have seen, like, as we added in the video as well, we used to look for rotifer using microscope.
Yes, rotifer people have seen. Others, yes, Ritik.
Moina.
Please unmute and speak.
Yes, sir. Nematode people has also seen in the cube lab.
Yes, so nematode people have used microscope and rotifer people, and I know fruit fly people. Yes, Moina. Okay.
Fruit fly people also have used it.
Okay. So fruit fly, Moina, nematodes, and rotifers. So how many of you have used this during the lockdown?
Nobody, because we didn't have any microscope as such.
Yeah, but what all did you try doing?
So from fruit fly group, actually, I think Ashit is not joined yet, but
yeah,
we were using a phone camera to. So actually, yeah, she did it using a phone camera to see the wing pattern, because that's how we identify whether the fly, the family of the flies, fruit flies. We
Yes,
were trying to identify Drosophilidae family. So yeah, that's what we were doing.
Yeah, so Aswati has also joined. Can Aswati tell us, like, what are the things that she managed to do with mobile phone and what are the things that she didn't manage to do? Aswati?
Yes, sir. So in our home lab, what we are doing that to make a microscopy, which are available at our home. Okay, sir. So we need a first we are doing that to take a photo from our mobile camera directly using flashlight.
Okay.
And maximum zoom in the photo. Sorry, the phone. Sorry, sir. Yes, maximum zooming the phone camera. So,
Okay.
but yes, sir. What's the problem in that photo is not much clear. I mean, the light source is very less in that photo.
Okay.
While we taking time.
Okay.
So along with Ashwati, several other people tried Siddhi, Shipra, Yash, and we didn't get, of course, like the first photo was definitely not good enough. But
yeah,
it was like we started building upon things, as we say, in the discussion we had several ideas from several people from different parts of the country, and depending on the suggestions, we tried things out. Some worked, some didn't, and that's how we develop. It's not like one day thing. Yeah.
Yeah. So what is the minimal size of thing that you could see with good enough clarity using a mobile phone. So what is the smallest thing that you could see in the lockdown using your mobile phone? Yes, Aswati?
Yes, sir. So we are using but our objective is to identify Drosophila melanogaster looking their morphological characters,
Okay,
especially
yeah,
wing. Yes, sir.
yes.
So we want to their wing to identify their wing vein pattern. We want to see their veins clear at our mobile phone. I mean phone.
Okay,
Sorry,
okay,
taking
okay.
phone picture.
Also, like Hina ma'am was adding that the photos of mosquitoes are all clicked by the students by using the phone camera and not really any other thing. So yeah, we could see the photo of mosquito, and which is quite clear, I must say.
Okay,
Yeah,
so
but at some times while I click the photo, to
Mhm.
zoom in more, the pixels of the photo generally break down very badly, and it gets too blurry for understanding the
Yes,
mosquito.
yes,
Yes.
right. You're right.
So
We can see,
yes,
hello,
Batul. Yes, Batul.
we can see size of 1 millimeter also. We can see the nematodes using the home setup of a torch and slide with a mobile phone on top, which
Okay.
Aswati explained.
Yeah,
We
yeah,
can
yeah.
even see the break in the wing. The break in the wing is very small, so that can be clearly seen using the home setup.
Okay, break in the wing of the fruit fly,
Those
right?
fruit fly, fruit
Yes,
fly.
yes.
Yes. So I am just writing down fruit fly, nematodes, moina, and what was the other organism? Rotifers.
Rotifer.
Okay. So looking at all these things, like from microscope point of view, like what can we say about the question that I asked earlier? What is the minimum size of the, what is the smallest thing that we can see?
In the wing and moina, almost 1 mm size, I guess.
Okay.
I guess 1 mm. Okay, 1 mm.
Around 1
And
mm.
rotifer, yes.
Yeah,
Rotifer
around
is
1
0.
mm. That too blurry, I guess. Little bit.
Yes.
Okay, okay.
At the highest pixel.
Can we, so are photos of these things there on Meta Studio? Because Ravi is anyways going to open it, so we can see some pictures there, because we don't have these photos readily right now. All the presentations would have gone. I don't think the presentations will be here. What are the photos that are available on Meta Studio? Does anyone of you know, so that we can
My
quickly?
mosquitoes photos are there if you want.
Okay, okay.
On Meta Studio.
Okay, okay. So Ravi, can you quickly open it, and in the process they will also sort of see
Okay,
the
so
searching
should I?
process. Yes.
So I will open the chatShaala interface. Do you remember
Yes.
the name of the thread? What is it? What was the title of the?
Yeah, let's search
Okay,
that. Ravi, you can start screen sharing and we'll search.
Okay,
Yeah.
so let's go to Cube.
Okay, so Rahil, where do you think we can find? Do you remember the name, or if we
You
search?
just search mosquitoes,
Very
something
good.
like that. Mosquitoes, you can search.
Very good. Okay.
Yeah, the top four all are.
Okay, so there are one with 17 posts. Okay. Okay, so all these are taken using mobile phone.
Yes, yes. Sir.
Right. And we also have some pictures from other. So just keep an eye on how big these are. Did you write anything about the size anywhere?
Of the pixel or the zooming?
Size of the organism as well as the zooming thing that you have done, like how many times you magnified.
Zooming, maximum I have to do 3, because if I get near the mosquito, it will fly.
Okay, 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 okay.
Maximum 3.
Okay, okay. So when Rahil is saying 3, I'm assuming he's saying 3 times, 3x kind of a thing.
Yeah, 3x, 3x, yes.
Okay. So let us look at some other organism. Are nematode pictures here? If we want to quickly see. Moina is there. Okay.
Yes,
Rishtaf has
nematode is also there.
Okay, nematodes are also there.
The one which is for filtration method, the one where is the filtration method, you can see the 1 millimeter size of the nematode.
Okay.
Nematodes.
Yeah, so I think Moinas we just have seen in the intro video as well as just now, along with the scale we got good enough photos.
DP, there's another photo of Moina recently. I have just posted 2 minutes ago. You can check on that.
Okay.
Some of them are good, showing spots.
On which thread can someone?
Yeah, the same thread.
Go down.
Yeah, the latest photo. I mean the latest post. Sorry.
Latest post. Okay. So I will go to maybe search from home. Yeah.
Yeah, the first one itself is
this one. 8x.
Okay. So 8x via digital zoom. So this is the picture that you managed to take, and nematodes were also taken.
Yeah, to isolate nematodes from home soil.
Yeah.
From home with using in the soil. Yeah.
Yeah.
From soil at home.
Yeah. Can you search that as well, Ravi?
To isolate, right? Maybe I can.
Nematodes group video. Go to home cube.
In the cube chat?
Yeah.
Okay.
Yeah, Kunal has shared us the link. So search nematodes from soil. We just saw that a while ago.
These are the posts which are popping up. Maybe if I just type soil. Is it posted inside the cube chat or was it?
It's in cube chat.
It's in cube chat.
And how when it was posted? Was it a recent?
September 17th. September 17th.
Okay,
it's
then
on
we can
September 17th. Yeah.
just go through the topics.
Okay, so basically, meanwhile, like when we look for that photo, the idea is to get a sense of what is the best that we can do with the mobile phones that we have. And I'm pretty sure all of you have tried a lot of things, and when you try to pick these pictures of whether they are nematodes or mosquitoes, as Rahil has done and others, so there are pretty good photos, and we shouldn't forget the classic picture that we always keep looking at by Aswati. She has taken a lot of versions of the nematodes. Sorry, not nematodes, of fruit fly wings, and the size that we see is almost of millimeter level. Slightly less than a millimeter can also be taken to get a good enough picture. Yes, Arun, you wanted to say something? Yes.
Yeah, DP, that mosquito photos of Rahil or somebody.
Yeah.
Is a very interesting thing. Some nice photos are there where the black and white spots can be distinguished very well.
Yes,
so
yes,
that means
Aedes mosquitoes.
It also tells that spot will be very small spots. So we are able to distinguish. So that also talks to us about the resolution. So I think one of those photos, I think we'll have to see it a little more closely. I'm sure other photos also will be, but this will be very black and white. So very fine.
Will be very clearly.
Yeah, no, fine resolution you can see there. No, good resolution you can see there. So let us take one or two photos of that, one photo of that, and look at it how neatly that we can see the small spots.
Okay. So, Ravi, can we get back to the mosquito thing? And okay, I can also do that.
This is also true with some of the photos of cardamine. I remember some people have taken the cardamine. There are some small, very minute spines on the leaves.
Yeah.
So some photos shows up that very well, which will be very small.
Yeah. Ravi, can you get back to that picture on mosquito?
Yeah, just a second. Oh, is a sharing.
Yeah, it's visible.
Sir, go to the second search result. Yeah,
This one? Okay.
Second or the I don't know which number was that where we saw AD's alpha pictures.
With
we see some stripes on the legs of the mosquitoes.
Even on the head, you can see a single line-like structure.
Yes.
Yeah, but the spots will be very interesting. You know, there are some very few photos which look at.
Yes.
There is one more better one. So let us just zoom it.
Scroll down. Yeah.
Should I scroll down or should I zoom this one?
Yeah, just zoom it and show that. So let us understand. Yeah.
I think
so. Sorry, Rahul says that this is
This is Aedes aegypti. Sorry, Aegypti.
So Rahul, my question is that what do you think is the size of that white spot and the black in between? What will be the size of that?
Sir, very small. If we
yes,
say
that's
that
what we want. How small? What is the size? What is the size of the mosquito? Sorry.
Size of the mosquito is 3 to 6 mm.
Okay. And what would be the size of that white spot that we are seeing if this entire thing is 3 to 6 mm?
It will be 1/16th, I feel, because it is on very small areas, in small parts only. 1/16th or 1/30th, whatever.
Okay.
But very small amount will be there.
Okay, so if I take 1/15th of 6 mm, that's how much? 60. It's 0.04.
Okay, 0.5. What is that? 0.04.
0.4 mm.
Okay, 0.4 mm.
Yes.
The size of rotifer, so we can try find rotifer with the phone. Like, this is giving me inspirations.
So what is the size of rotifer? What's the size of
rotifer is 0.1 to 0.5. It varies from 0.1 to 0.5. So yeah,
take it as 0.3 or 0.4.
Yes, if you can see 0.4 dots on Aedes aegypti, then definitely see a 0.3, 0.4 animal. Yes.
Yeah, the point that is a guesswork of Rahul. Okay, so if we go more close, I think we'll be able to see exactly what is the magnification that we are able to get from a very casual photograph. You know,
yes.
I'm saying in the sense we have not random photograph that he has taken, and some of them are good, some of them are not so good. But see the resolution that you can get. You know, that's what I think we need to look at.
Yeah, but I think it is here. I just want to tell a few more things that are important when we are taking photographs. So there is something called still photography, in the sense the mosquito here is very stable, at least stable within the time scales of the photo that Rahil has taken. Whereas when we take up rotifer or very small organism, which is moving, you will be able to still see the organism definitely, but the clarity might be not as good as, might not be as good as what we are capturing here. But those are all the various sort of, if you remember, like the discussion that we had when Farhan talked about the oscilloscope as well. So he was very specific about the limits of his instrument, which is an oscilloscope. Like what is the range of the signal that it can measure. So the range there is the voltage and the frequency. And in the same way, for photographs as well, there are certain parameters that we need to consider. And I'm not going to tell or give a lecture on what all those parameters are going to be and all those things. But essentially, what you have been trying out is basically tinkering with your mobile phone's camera and trying to get good enough capture of the organism that you're interested in, right? Whether it's a mosquito or a fruit fly's wing or a nematode or a rotifer, right? So these are all really beautiful pictures that you're able to capture, as audience in YouTube as well as here are saying. But we need to identify those boundaries of our mobile phones. And to be able to do something in the spirit of home lab that we currently have, because of the lockdown and others, I think we will also need to take this to the next level. Once we identify like what is the best that we can do with our mobile phones, we'll have to identify like what are other ways of extending the mobile phone, and two, building other microscopes within our home labs. So I think that is what Ravi was trying to show in the beginning. Ravi, can you get back?
Yeah, sure.
Yeah. Yes, so there are lots of ideas that we, all of you have been having. So what we are planning to do is we are planning to have a series of discussions because it's not going to happen in one session in half an hour or 40 minutes or even 1 hour. So we need to engage much more closely and do certain things so that we identify where all our mobile phones are going to be useful and what can we do for other purposes, for things where we need photos of, say, higher resolution. We need photos of higher magnification. We need photos of slower pace. If someone wants to capture the wing of a mosquito when it's flying, so there could be some issues there. So we need to identify those limitations and find out how we can build those things. So as an exercise now, before we end this session, what I request all of you to do is there are some posts that are already there on chatShaala.in. So this way, like, you can also get familiarized with chatShaala.in. So you just go and search microscope and see what all things are already happening and write your responses in any of the threads down there. Okay. And what we will do is, again, in the beginning tomorrow, we will come back and have a quick conversation for 10-15 minutes, just the way we are having right now, looking at those responses. And then we will plan out the series of activities for the next few weeks to months, where we will go beyond what we have been doing right now. And yes, so these are all things that people have already tried doing just the way you have done. So you can write down your experiences when you tried to magnify the pictures in those threads. So Ravi can just show some of them. Maybe does anyone want to add anything so that we can post it right away to also show once? Iske niche.
Yeah, I think what we can basically do, DP, is like let's create a separate thread in which we just collate all the approaches which people have tried like so far
Great,
to
great.
capture the micro world
Yes,
around
so
us.
let's
And the
show
second
how to
part
create
would
a
be
post. Yeah.
yeah.
So,
the second
I
part,
can suggest one small challenge for everybody.
Yes, GM.
All of us have different kinds of phones, right?
Yes.
So one simple exercise that I want to do is, can you create a thread now and ask this question, a new topic under Cube chat, category Cube chat. The question can be something like, find out what is the focal length of your phone and post the result. And how did you find out the focal length? How did you measure the focal length? A very simple task. Because this is very important for us to find out how to make your phone into a microscope. So, that's okay. And in the description, I would like to add a few more things. So in the conversation, please write, find out what is the focal length of your phone. Yeah, find out the focal length of your phone. And two, one more question I want to, and also tell us how you found it out. And the third question is, what is the smallest area that your phone can capture?
So you can also post your pictures like when you're.
And these are the important questions, and once you know this, then you will be able to find out the limits of your phone as well.
Yes.
That DP was talking about.
Jane, can we also make...
Yeah, one more question I want to ask. The fourth question, can you add the fourth question also?
Yeah, sure.
Try 2x 2x, that is 2x zoom, and let's say 4x zoom, and measure the smallest area of the phone can capture. That is answer 3 at different zoom levels.
Yeah, yeah.
Okay, so let this be the question. Maybe by tomorrow evening, let us get some answers, and then we can continue discussing this.
Yes, yes, yes. And also please
and also share the pictures. Yeah,
share the pictures and use this as an opportunity to also become familiarized. You can try out registering things. So in this process, you'll also figure out if you are facing any glitches and others. You can inform us during the day as well as before we leave tomorrow. So the post is there now. All of you can reply there just the way you reply in your WhatsApp or Telegram interfaces. You can attach pictures there. And we are going to discuss these things again tomorrow. And we will continue these discussions after we identify the limits of our mobile phone towards building a home microscope for ourselves. Okay. Is there anything else that you want to add, Ravi, or others you want to say?
Yeah, I think tomorrow, most probably Jude and Ashish will also join. Like they have worked, they have worked on the initial design of the the web camera based microscope, which I was showing earlier.
Yes.
So we'll also get to like listen to them like about their design and their approach, what they did.
Yes.
Plus, do look at the, as DP shared, we did a couple of offline workshops, one at Sherpur NMS and also at HVCSC, where the participants made different designs, which I was like sharing. So I think those could also be an a trigger to
yes,
kick off new ideas.
And if any of you are facing any glitches, that shouldn't deter you from posting. So I request others to help convey your responses through your posts until they can smoothly get into the chatShaala interface. Yes.
So my suggestion is like what you said. That first let us see that how we make a home lab microscope.
Yes, yes.
With whatever possibilities that we can make use of with the mobile camera.
Yes.
So because the other things, gadgets, any other gadgets that you will have to buy it from market and get it and all that, I think it will be only some people can do it. So what is the lowest minimum we can use in order to get the best possible thing? That's the first chapter.
Yes, yes, yes. So I think that is our first objective. If we use the language that we, or short-term objective or short-term goal kind of a thing, to identify what is the capacity or what is the limit of the device that we have in our hands in capturing the photos that we want to in of small objects especially.
Yeah. So DP, I have one more additional thing. See, for example, everybody will think that we have very limited scope with our whatever we have. So that is what we have disabused. We have changed it when the Drosophila photography was done.
Yes, yes.
Yeah, and that too when it was done, some people got a little better, some people got good, better ones. But what is it that makes it better? Is it by chance or is it that there is science behind it?
There is a technique behind it. Yes.
Yeah, technique and also science, because where the light comes from, I think we did a lot of initial analysis of that. No, not a very meticulous one, but so where should the light come in order to project or in order to get a better resolution of a particular spot or something? Two spots. Yeah, two lines, very two narrow lines. So how do we get that resolution? And for that, what type of lighting that we need? And now I can assure you that Ashwati, Siddhi, Shipra, etc. has done, including Yash, several such things, and even the school students joined in order to draw the optical line drawing.
Yeah.
So what type of light gave a better result? What type of whatever? So that's a very, because otherwise we think that this is all what we can do. So that's also why today I showed you that the fine difference in the mosquito.
Yes.
Yeah. So if you can see that, okay, what is it that we have been able to magnify there, and what was the resolution that we got there by a random shot by Rahul, which essentially means if that random shot can be standardized or calibrated, that means we can improve upon that.
Yes. So that's the thing, and we even if you do not understand or are not clear, because when the moment you heard focal length, the moment you heard resolution, various other things, you may think, okay, this is not my cup of tea, I'm not going to do. So don't do it that way. Whatever, just take it as a task, and a minimal thing of taking the smallest object that your phone can pick up. Tinker around, try various things, keeping objects close enough, keeping objects far enough, making objects small appear bigger, smaller. Try various things, try something and post a picture there so that we can talk. Otherwise, there will not be a context for us to for you to connect at all when we discuss. So I think that's what is very necessary. So
yes,
yes, Ashutosh. So we are hoping to have a lot of pictures, and we'll continue this conversation tomorrow again. Over to Ashutosh.
Yes, certainly. So this particular activity is really nice. If we could have people participating in it throughout the day tomorrow, and even when we join the session tomorrow at 5:30. Yes. So so that's all for the day today. We had very good discussions. See, people seem to getting the getting into the tempo of it. So yesterday, so we had a many more discussions today as well, and we are looking forward to even more discussions in the tomorrow's day, the final day. And we certainly have a many more project work to talk about. So in tomorrow's day, we would have uh, um, people talking about phyllanthus plant, work on phyllanthus plant, earthworms, zebrafish, hydra. So many activity tomorrow as well. Looking forward to it. That's all for today. Looking forward to for you to again join tomorrow. Get in more people to join tomorrow. So see you there tomorrow at 5:30.
Yes.
Yeah. Bye.
Yeah, I think we can stop the stream now.
Yes.
Okay, thank you. Good night.
Arunan, we also had to identify a chair for